Alright, what's up everyone? I just want to check my audio before we start, so please leave in the live chat whether the audio is good here. I'm trying to work out the kinks since I am new to live streaming, so if you guys could just let me know if the audio sounds good in the chat section. Okay, cool. So before we begin, I just want to say, just as a little disclaimer here, I don't have a position in BNGO stock. So what I'm trying to do here on this channel, and a lot of people are going to say I have a bearish take on the stock, and to some degree I do, but at the same time I'm trying to give the best information that I can. So that's the entire reason why I'm holding these live sessions that I am. Just so the science can speak for itself and you can decide on whether or not you want to buy. So I'm not going to tell you whether to buy, sell. As many times as you ask me in the chat, I'm not going to be giving my opinion on that. I'm just going to let the scientists speak for themselves, and you guys can come up with your own conclusions. So that's just my 2 cents on it, and I hope you enjoy today's live stream. Thanks for joining. So there doesn't seem to be any audio on this presentation yet. If any of you guys are actually tuned into the web seminar on their website or on event24.com, let me know if you guys can actually hear anything, because I'm not hearing anything at all. Actually, I'm just going to try to play a clip from the last symposium just to see if the audio is working correctly. And I'm sorry if there's any hiccups with the audio. Again, I'm new to streaming, so hopefully you guys can bear with me here. So it seems like my audio should be working, and they're about to come on right now. So I'm still not getting any audio output from them, but I'm not sure if they have their sort of elevator music that they usually play before these symposiums. Let's see. Okay, so I'm actually gonna have to go back through this. For some reason I'm getting an error. Sorry for the technical difficulties again. I'll try to fix it as soon as I can. Let's see. Your registration cookie is not present. Okay, well that's not gonna work. Well, I had it going before, and now this wants to be temperamental with me, so bear with me here again. Alright, here we are. And let's see if I'm getting any audio output from my OBS system. Okay. So you guys should be able to hear her. Let me know if you can. And now this is going black screen. I'm not sure what's wrong with my computer right now. There we go. Let me fix my nice setup, see if I'm missing anything here.
As of today, for hematological malignancies, using recommendations employed globally by World Health Organization include cytogenetic testing using karyotyping FISH and CMA or chromosomal microarray, and also molecular genetic testing for targeting sequencing variants. Also, National Comprehensive Cancer Network, College of American Pathologists, American Society of Hematology also provide guidelines for appropriate laboratory testing for the initial diagnosis of hematological malignancies, which includes, again, karyotyping, FISH, or fluorescence in situ hybridization, chromosomal microarray, and also sequencing variants of targeted genes using molecular genetics. So why is cytogenetics or cytogenomics in neoplasia important? Because there are unique structural variants which are associated with each leukemia or lymphoma subtype. And these structural variants include translocations, profile of those translocations can be better characterized. So structural variants can be among the hardest to detect genomic variants. We have sequencing technologies and other orthogonal methods that can reasonably detect single nucleotide variants at base pair resolution. And we also have conventional methods which allow for the detection of whole chromosome aberrations such as aneuploidies. However, when we look at structural variations in the different classes, which I will go in detail in a bit, which range from hundreds of base pairs to millions of base pairs, and each technology employed globally as of today as standard of care falls within this range with its own limitations. This is a slide courtesy one of our collaborators, Dr. Eddie Maher from the United Kingdom, where in his cytogenetic laboratory, you can see that not only are the cytogenetic methods complex, involving extraction, culture, performing different assays, but along with the complexity, these methods are also slow and require integration of a significant amount of data from karyotyping, chromosomal microarray, and FISH to result into a diagnosis. On the right-hand side is our adaptation showcasing how the optical genome mapping workflow is extremely simple and user-friendly and cost-effective for any laboratory. Sequencing technologies perform poorly for the detection of structural variants. The short-read sequencing technologies for large structural variants have demonstrated poor performance. But if we look at long-read sequencing technologies, they have inconsistent throughput, are not cost-effective, and if we compare that with optical genome mapping, not only do we have the right throughput, but also we have a cost-effective technology which performs at approximately 5% allele fraction at 400x step and can be as low as 1% allele fraction at 1600x step. This is extremely important for looking at different clones and subclones in leukemias, lymphomas, and other types of cancers. So let me introduce all of you to optical genome mapping technology with Saphyr. What you see on the left-hand side is this beautiful video, which is going to show us how these long linear molecule of DNAs are imaged using the Saphyr instrument, while these DNA molecules are shooting through these nanochannels on our Saphyr chip. In the bottom panel, what you see is the images of these molecules where the backbone is counterstained and these green dots demonstrate the specific labels based on which the pattern recognition of each chromosome will take place using optical genome mapping. So optical genome mapping has a very simple and single workflow, where for hematological malignancies, you can start with 650 microliters of blood or bone marrow aspirate, and we have special protocols for isolating ultra-long DNA molecules, and we use a special enzyme to apply the label pattern onto these molecules, which are then loaded onto the Saphyr chip. As I showed on the previous slide, those molecules are linearized and are pushed through these nanochannels and then are imaged. And we have the high throughput, high resolution imaging of these ultra long DNA molecules and are aligned to a reference genome to detect structural variants. So structural variants are detected using optical genome mapping by comparing the patterns to a reference. In the green bar, you see the reference panel with these different patterns, and in the blue are the sample Bionano maps, which are used to compare and detect structural variants. So a deletion results from the loss of a few labels or the decrease in distance between two labels. And an insertion may result by introduction of additional labels or increase in the distance between two labels. Similarly, repeat array expansions can be adjudicated based on the pattern recognition, and also duplication of a certain region of the genome just based on its pattern can be determined, and also we can determine if the orientation is tandem or if they are reversed. One of the most important SVs to detect and highlight for hematological malignancies is the ability to detect and identify these unique translocations where the patterns will map to two different chromosomes, which allows us to determine which chromosomes are involved. Also, we can pick up inversions just based on the patterns and the differences between the reference and the genome of interest. Along with our structural variant maps, we also have an algorithm which gives us the copy number variant profile, where you can see chromosomes 1 through 22 and X and Y, which allows us to pick up whole chromosome copy number gains and losses. If we look at the structural variant classes, if we focus on aneuploidies for monosomy of a chromosome or trisomy of a chromosome, these can be very easily detected by optical genome mapping. For copy number variants, whether it is deletions or duplications, we have the ability to pick up interstitial deletions, terminal deletions, and for duplications, we can identify tandem duplications, inverted duplications, or if those duplications are inserted anywhere else in the genome. As I mentioned previously, insertions of known or unknown sequence just based on the patterns can be very uniquely detected. Now for translocations, certainly balanced rearrangements or balanced translocations and unbalanced translocations can be very easily detected, but also cryptic translocations, which are beyond the limit of detection of the current technologies which are used as standard of care, can be very easily identified. Also, inversions that are pathogenic and that are associated with a specific leukemia or lymphoma can be detected accurately. We do not detect single nucleotide variants. So in this slide, I want to orient everybody to what you will be seeing in our amazing list of speakers for today. So all the structural variants using optical genome mapping are presented in a circus plot where the outer ring demonstrates or shows the ideogram of the entire genome from chromosomes 1 through 22 and X and Y. The next ring actually shows different colored dots, indicate whether they are insertions, deletions, inversions, duplications, or translocations. The third ring shows the copy number variation profile, and the inner ring demonstrates if there is a unique structural variant, such as a translocation taking place. In this case, there is a translocation involving chromosomes 11 and chromosomes 22. Also, we have the ability to filter all of these variants against our population control database, or also compare a tumor with a normal. And the users also have the ability to filter these variants against a bed file of their interest to identify structural variants. So with this introduction, I hope that all of you will remember this, and as our speakers showcase their amazing data, you all can appreciate how useful and beneficial this is. We have an amazing speaker lineup today, followed by our panel discussion and Q&A. Please do not forget that, and join all of us again at the end of our session today. We will start with Dr. Tuomo Manter from Lewis School of Medicine, showing the studies on 52 leukemia genomes by optical genome mapping. This is followed by Dr. Blanca Espinet from Hospital Del Mar, where she will be speaking on the genomic complexity in patients with chronic lymphocytic leukemia. This is followed by Dr. Gordana Raka from Children's Hospital Los Angeles, where she will show rare and genetic drivers in pediatric BALL cases. And then Dr. Rashmi Kanagal-Shamana from MD Anderson will show her data on the clinical evaluation in myelodysplastic syndromes. This is followed by Dr. Valentin Lestrignan from CHU Amiens Picardie, where we will see optical genome mapping used in a series of acute lymphoblastic leukemia cases. And then Dr. Hongjin Liu and Dr. Bo Chen from Liu Dopei Hospital, where we will see how they used optical genome mapping to decipher structural variations in acute leukemias. We will have Dr. Barbara Devale from UZ Leuven, who will again showcase her work using optical genome mapping for the genetic diagnosis in acute lymphoblastic leukemia. And we will end with Dr. Bryn Levy from Columbia University Medical Center, where he will show the work as part of a national multi-center evaluation using optical genome mapping for the assessment of structural variations in acute myeloid leukemia. Please stay tuned for all of these amazing talks and join me at the end of the session for our live Q&A. Thank you very much.
Good day, everybody. So my name is Tuomo Mantere. I'm a postdoc. I worked at the Immunogenomics and Genomic Technologies Research Group at the Radboud University Medical Center in Nijmegen in the Netherlands. And currently I'm working at the University of Oulu in Finland in the field of cancer genetics. So today my talk will be about the cytogenetic assessment of 52 leukemia genomes by Bionano Genome imaging. So we obtained the Bionano Saphyr system in Nijmegen in early 2019 and had the training in March 2019. And since then we have ongoing research projects, mainly focusing on detection of somatic structural aberrations in leukemia, and compared to the standard of care diagnostic test results, including FISH, karyotyping, and CNV microarrays. And then we have another ongoing study in collaboration with our dear French colleagues, and they will be, this is about the detection of structural germline cytogenetic aberrations, and there will be another presentation by Dr. Laila El-Khattabi on this. And finally, our third approach has been to use Bionano Genome imaging to solve some of these really difficult to solve rare disease cases. And there is another presentation by Dr. Alexander Hoyshen on this topic in the same symposium. So, as many of you know, the comprehensive analysis of all structural variants in a given leukemia sample, it still requires a combination of different techniques, including traditional karyotyping, FISH analysis, and CNV microarrays. So in a diagnostic setup, it would be highly beneficial to have a generic single cytogenetic test to use instead of all these, the combination of all these three. So that in mind, we set up proof of concept study where we included 52 leukemia samples that were analyzed with the standard of care diagnostic cytogenetic tests, including microarrays, karyotyping, and FISH. And these 52 leukemia samples, they consisted of a mixed bag of different types of leukemias, most commonly MDS and AML. And the leftover material after the standard of care tests was used for genome imaging with the Saphyr system. And then we, based on the diagnostic test results, we divided our cohort into simple cases which had less than 5 diagnostically reported aberrations, and to complex cases which harbored 5 or more aberrations or unspecified marker chromosome. And the main source of material in our cohort was bone marrow aspirates, about 80% of samples, and then rest were from peripheral blood samples. And we always isolated the ultra-long DNA molecules from 1.5 million cells and performed the labeling using the latest DLS chemistry. And the samples were then run with the maximum capacity of 1300 gigabase pairs of the current chips that we are using. And then a little bit about the technical summary for our study cohort. So indeed, we had about 1300 gigabase pairs of data per sample, and the molecule length, the length of the molecules that were used for the analysis on average was 260 kilobases, and the map rate about 72%, resulting in effective coverage of 280-fold per sample on average. It's really high coverage. And as many of you here might know, that there are two different analysis modes in Access to analyze Bionano data. So the de novo assembly-based variant annotation and SV and CNV calling. So this is what we use for germline constitutional variants. But in this study, in this leukemia study, we used the rare variant pipeline for SV and CNV calling, which is specifically meant for detection of somatic structural variants. And so how much do we find SVs and CNVs per sample is a question we often encounter. So on average, there is 13 high confidence SV calls that are larger than 100 kb per sample. And this is after excluding all the common germline SVs using a population control database. And 10 of these SV calls are overlapping with a gene. And quite many of these calls are from result from large rearrangements such as interchromosomal translocations or large inversions or interstitial deletions. And for CNVs, there is a separate algorithm calling CNVs. So on average, there is 8 gains and 47 losses per CNVs per sample, called as loss. And some of these large, this number may seem rather high, but it's good to note that some of these large CNVs or aneuploidies are over-segmented. So actually, there's like one single big CNV is called as multiple different ones. So these numbers are somewhat inflated. But of course, we also have really highly complex and rearranged samples that do harbor a lot of CNVs. And then the most important question is that how are we concordant with the standard of care results? So are we able to find back all the aberrations that were previously picked up by the diagnostic tests with the classical approaches? So I will start with the simple cases. We had 36 simple cases, as I said, and these harbored in total of 46 aberrations that fall within the scope of this study. So for now, we had to exclude the copy-neutral loss of heterozygosity and structural variants, that balanced structural variants with centromeric breakpoints, and structural variants with the variant allele fraction less than 10%. But in total of these 46 aberrations, they were most commonly we had deletions and different types of translocations, but also aneuploidies, inversions, and duplications. And important result here is that we find them all back. So we reached 100% concordance for the simpler cases. We didn't miss any of the previously detected aberrations using Bionano genome imaging. And for the complex cases, we've reached the full concordance for 15 out of 16 of these cases. And one of these cases that which actually had multiple different aberrations, there were three CNVs that we were not able to pick up with genome imaging, but all of these aberrations actually had variant allele fraction around 10%, which is just the borderline of what we are able to detect with the current setup. And generally, for the complex cases, it seems that the optical mapping is able to more fully resolve the true underlying genome architecture compared to the previous tests alone. So I will show some example later on this. And in addition, we are able to find a lot of new aberrations that were not picked up with the previous tests. For example, balanced translocations. And this opens up a lot of possibilities for new research as well. So then I will move on to some examples. So one important class here are the simple, in this context, small deletions that are hitting a well-known leukemia gene. So, for example, previously in this sample there was a 700 kb deletion hitting the TET2 gene picked up with the CNV microarray, and we run this same sample using Bionano. So in this circus plot view, you can already see that there is, this is chromosome 4, so we have a CNV and an SV call hitting the TET2 locus, and when we zoom into this, we can clearly see that this deletion is called by both the coverage-based CNV tool and also with the SV, the assembly-based SV tool. So both of the algorithms are able to pick this up nicely. And then moving on to one of the really strong points of Bionano, and especially important class of variation in leukemia, these are the balanced translocations. So in this complex sample, using a combination of karyotyping and FISH, they detected a translocation between chromosome 11 and 14, which results in this well-known IGH CCND1 rearrangement, which is commonly seen in leukemia. So we run this with the Bionano, and in this circus plot view of chromosome 11 and 14, we immediately picked up this translocation between these two chromosomes. And when we zoom into this event more closely, we can right away see that the genes IGH and CCND1 are involved in this rearrangement. So this is nicely concordant with the previous tests. And I mentioned earlier that we think that we can find a more complete picture on many of these complex samples when we run it with Bionano. So for example, this same sample I showed before had a multiple CNV calls with the CNV microarrays here. For example, these gains and losses of chromosome 3 and chromosome 8. And when we run this with the Bionano, we can see that in quite many of these complex samples, we see that the findings from the previous CNV microarrays are mediated by previously undetected translocations, as here. We can see that the gains and losses in chromosome 3 and 8 these breakpoints are matching exactly with the breakpoints of these translocations that were previously undetected, despite the use of karyotyping. So moving on to a chromotripsis case, we had this sample with the karyotyping, with the combination of karyotyping and CNV microarrays, we're able to pick up there's a chromotripsis of chromosome 8. And once again with Bionano, here with the circus plot view, we can see that the circus plot is quite clean for other chromosomes, but for chromosome 8 it's just full of large rearrangements and complex deletions and inversions and everything. And the CNV profile is also, if you look at this figure, it's matching quite nicely with the CNV microarray on top and the Bionano CNV profile on bottom. And it's matching really well with what we see with the CNV microarray. And then this is a well-known recurrent translocation between chromosome 9 and 22, resulting in the BCR-ABL fusion, which is a commonly seen aberration in CML. So in our cohort, we had 3 CML cases with this BCR-ABL fusion, and with Bionano we were able to pick up all these 3 events. And on the left we can see that there are 3 different predicted breakpoints in ABL gene and 2 different breakpoints in BCR. So it seems that the breakpoint resolution is much better than in traditional fish also. And then one additional thing we did for our samples was we tried to look for balanced translocations that could lead to potential novel fusions. So what we did, we checked for all the balanced translocations and that are involving a known cancer gene from Cosmic database, and which were not reported in our dataset with the previous techniques that were used. So we identified some of these as well. One of them was this IGH-BCL3 translocation involving these 2 genes. This is actually known in literature, but it was previously undetected in this sample, and it could be really clinically important translocation as well. And then there is one other MSI2 UBE3C translocation involving these 2 genes. And this is actually really interesting for research, since this was the only aberration that was detected in this sample, and it might result in a fusion, novel fusion that could be potentially a novel driver of leukemia. But of course, this requires a lot of follow-up still. But overall, I would say that Bionano can be used as an efficient tool for mapping novel putative fusions. And then also about the copy-neutral loss of heterozygosity detection. So currently this is not yet enabled in the tools that Bionano offers, but we manually take a look at this sample where we knew already that there is a large region with the copy-neutral loss of heterozygosity in chromosome 7, and we just manually checked the zygosity of all the SV calls in these regions and compared this with the region that is not supposed to have a loss of heterozygosity, and we can quite easily see from here that the heterozygosity is lost in this sample, in the matching the CNV array results very nicely. And then I would like to conclude, just summarizing a little bit. So we had 52 leukemia samples, and these had previously been tested with the standard of care cytogenetic tests, including karyotyping, FISH, and/or CNV microarrays. In total, this consisted of 36 simple cases with less than 5 reported aberrations, and then 16 complex cases with 5 or more aberrations. And for the simple cases, we were able to find all the previously reported aberrations with really low false positive rate. And for the complex cases, for 15 out of 16, we found all the aberrations back. For one case, we missed some aberrations with low variant other fractions. But overall, the complex cases seem to be even more complex than previously thought based on the previous tests. So the genome imaging, it's likely closer to the true underlying genetic architecture in these samples. And the next step for us is to perform a prospective study for leukemia in early 2021, where optical mapping and standard of care tests are run in parallel and compared. And then I would like to acknowledge especially Cornelia Nevling and Alexander Hoyschen, who have been really the driving force behind this study, and many other colleagues. Also Marianne, especially for help in the classical cytogenetic approaches, and many other colleagues and the people from Bionano for their help in many aspects of this study as well. Thank you.
Hello, everyone. My name is Blanca Spinet, and I am cytogeneticist, and I am the head of the Molecular Cytogenetics Laboratory in the Pathology Service of Hospital del Mar in Barcelona, Spain. I will share this presentation with my colleague, Dr. Anna Puig-Gros, who is also a cytogeneticist, and together we will present our experience on the analysis of genomic complexity in patients with chronic lymphocytic leukemia using Bionano Genomics optical genome mapping. CLL is a mature B-cell neoplasm. It is the most common leukemia in Western countries, and the median age of diagnosis is 70 years. The etiology is unknown, and the median survival is around 10 years, but this disease can present highly variable clinical courses, and that's why the researchers have looked for prognostic factors to predict which patients are gonna go well and which are going to go very bad. One of the prognostic factors is genomic complexity. And this is a very old prognostic factor. In fact, it was already present in the seminal papers of Juliusson and collaborators from the 90s, in which all the cytogenetic studies were performed. And you can see here the survival curves of these patients, in which you can see that patients having 3 or more chromosomal abnormalities had the poorest outcome. The classical concept of genomic complexity was defined using cytogenetics, chromosome banding analysis, and until now, this concept is we think that a karyotype is complex when we have 3 or more cytogenetic abnormalities in the same cell clone. And you can see here an example of a very complex karyotype and the ISCN formula below. This concept was defined in the 90s in myelodysplastic syndromes and acute myeloid leukemias, and the concept in CLL was adopted from these informations. And this, in recent years, the importance of complex karyotype has resurrected. In fact, with the introduction of new drugs, new agents in CLL, this concept has arisen again, and we are now interested in see if the patients having complex karyotype perform better using new drugs such as ibrutinib or venetoclax compared to chemoimmunotherapy or allostem cell transplantation. In this sense, in this context, the European Research Initiative on CLL, called the so-called ERIC, has undertaken 2 major European studies to analyze karyotype complexity. The first paper was this one depicted here by Baliakas and collaborators, in which we also collaborated as a Spanish cytogenetics group and a Spanish group on CLL, in which we collected nearly 5,000 cases with CLL, from which 1,000 were complex cases, and we could analyze several issues. The second paper that Eric undertook was regarding the usefulness of genomic arrays to identify high-risk CLL patients. In this case, we collected more or less around 1,000 patients with survival information. And the third paper in which we have worked is the one that was organized by our institution, but several European groups have also participated, in which we have compared chromosome banding analysis versus genomic microarrays as two methods to stratify cases with CLL, taking into account their genomic complexity. And this paper is already submitted and it will be soon published. In this sense, the conclusions of these three papers are that genomic complexity is an important prognostic factor in CLL, mainly in patients treated with chemoimmunotherapy, but also in new drugs, but not all cases are equivalent. We have a very good prognostic group that is the multiple trisomies group. These patients having trisomies 12, 19, and other abnormalities. That low and intermediate complexity cases having 3 or 4 abnormalities impact only on outcome only if they have TP53 deletions and or mutations, and that high complexity cases having 5 or more abnormalities are associated with the worst clinical outcome independently of TP53 abnormalities and the IGHV somatic hypermutational status. And finally, we have described that both chromosome banding analysis and genomic microarrays are useful to detect genomic complexity. And in this context, we wonder if optical genome mapping was a good methodology also to detect genomic complexity in routine diagnosis. In this sense, our aims of this study were to analyze the usefulness of Bionano Genomics optical genome mapping in the assessment of genomic complexity in CLL patients. We wanted to compare the obtained results with previous chromosome banding analysis and genomic microarrays informations, and to correlate this information with clinical biological parameters.
To accomplish the aims that Blanca has commented, we assessed a total of 22 CLL patients with available CBA and FISH results that were divided in two groups, 10 patients who carried non-complex karyotype and 12 with a complex karyotype. Tumor DNA was used to perform genomic microarrays in 12 cases and optical genome mapping in the whole cohort. Optical genome mapping results were compared with those obtained with the traditional techniques in order to assess the concordance in the detection of known abnormalities and the complexity detected in both groups. Moving to the results, I will show the results regarding the complexity, the concordance detection of previously known abnormalities in each group separately. In the non-complex group, almost all the abnormalities were found, and the only discrepant patient is this patient, which shows an additional material of in the chromosome XP that was not detected by Bionano. The only abnormalities on this chromosome by Bionano genome optical mapping were a small deletion and 2 small insertions on that chromosome. I will show you also an example in which all the abnormalities were detected. It's this patient in which we detected a loss of chromosome Y and an inversion of chromosome 2 by CBA and an additional 13q deletion by FISH and also by microarrays. By Bionano, we detected the loss of chromosome Y. At this point, it's important to remind you that some aneuploidies in sex chromosomes are not called by Bionano access, and it's necessary to perform an initial review of the circus plot or the whole genome plot. The other abnormality detected was the inversion that was detected as an intrachromosomal translocation, and regarding the 13q deletion, it was detected by the rare SB pipeline. In addition, in this group, we found one patient in which the structural information provided by Bionano allowed the karyotype reinterpretation. It was this patient that shows a deletion 13q and an insertion of chromosome 15 on the 3q arm by CBA, and by Bionano, you can see here the circus plot obtained, and we found that the deletion 13q was more complex than previously assumed. It was composed by multiple deletions, and moreover, this chromosome was involved in several translocations with chromosome 15 and chromosome 3. With this information, we can conclude that probably the insertion detected was in fact a more complex rearrangement involving also chromosome 13. We are performing whole chromosome painting FISH analysis to confirm this hypothesis. Moving to the complex group, we only focused on those patients with genomic microarray data to perform a better comparison. And in this subgroup, we found that 85% of the abnormalities were correctly detected with highly concordant coordinates and with some additional structural information provided by Bionano. I will show you two examples of this group. The first is this case with a complex karyotype and clonal evolution by CBA, including additional material on chromosome 17, a 13q deletion, and a duplication of chromosome 1q. And moreover, by genomic arrays, we found a large loss and a large gain on the 3q arm. By Bionano, we confirmed the 3q deletions. It was found as an intrachromosomal translocation. We also detected the CMB gain that was detected as 3 large CMBs that could be joined. And interestingly, we found a translocation between the chromosomes 3 and 17 that when was assessed in more detail, we found that the breakpoint on chromosome 3 was just at the start of the gained region. So after the karyotype revisions, we conclude that the additional material on chromosome 17 was in fact an unbalanced rearrangement with chromosome 3. On the other hand, the deletion 13q was called by both pipelines in the Bionano Access software. The CMB pipeline detects 2 large deletions in chromosome 13q, and the SB pipeline detects an intrachromosomal translocation on these chromosomes. In both cases, the coordinates were highly concordant with the genomic microarrays, and with this information and other examples found in other patients, we consider that we could take into account the specific breakpoints provided by the SB pipeline and in the normality count, in order to avoid duplicities in the abnormalities, only consider those provided by the SB pipelines. It's important to note that despite we don't count the CMB losses, they allow us to confirm that the structural rearrangement corresponds to a large deletion. In this patient, not all the abnormalities were detected. The loss on chromosome 17 was a telomeric deletion found by genomic microarrays. In this case, as microarrays has a sensitivity of 25% more or less, we assume that the problem was not a sensitivity limitation of Bionano. And when we focused on the deleted area, we realized that in fact, most of the deleted area was masked by the Bionano pipeline. These masks are distributed along the genome, mainly in the telomeric and the centromeric areas, to avoid the high rate of positive, false positive calls. But in this case, we conclude that could be probably the cause of discrepancy. The other abnormality not found was a duplication on chromosome 1. It's important to note that in this case, genomic microarrays neither detect this abnormality, and our hypothesis is that it is a subclonal abnormality that is below the sensitivity of Bionano that has been expanded during the culture. The second example of this group is this patient with a high complex karyotype with multiple aberrations with material of unknown origin. Genomic microarrays also detected a high complex genome, including a chromatopsis event on chromosome 11. And by genome mapping, we detect that these patients carried a lot of abnormalities, and that most of the abnormalities detected by arrays were currently detected. Just as an example, I show you the copy number profiles obtained by genomic microarrays above and below those obtained by Bionano. And as you can see, the profiles are very similar in these two examples, chromosome 2 and chromosome 10, but also in chromosome 11 that shows chromodipsis. In this case, CMV losses were better detected than gains, but Bionano also provided us structural information regarding translocation associated with this event. Moving to the second aim, the assessment of genomic complexity. If you take a quick view of the circus plot of each group, the non-complex and the complex, you can see that the second group shows a lot of abnormalities and most of the patients carried a lot of translocations. But interestingly, when we compared the total number of FSVs and CMVs provided by the Bionano Access software, we didn't detect statistical differences. And these differences were mainly focused on the translocations and the CMV gains detected by the CMV pipeline. At this point, it's important to note that our results are still very preliminary. We are performing the data curations because we need to delete the duplicated abnormalities, as I have shown before, to join some fragmented CMVs and to filter some polymorphic SVs and CMVs that overlap with the DGB database. That this process is not automatic in our current version of the Bionano Access. And finally, as a conclusion of our study, we conclude that optical genome mapping in CLL is effective to detect most of the previously known abnormalities, providing also additional information in some patients, is a valuable tool to assess genomic complexity in CLL, and detected also many additional abnormalities of still unknown significance. Our future steps include the curation of the raw data obtained, to perform also additional genomic microarrays to better describe the cohort and FISH analysis to validate some variants, and finally to assess the genomic complexity in our cohort and correlate it with the clinical biological information of the patients. Finally, I would like to thank all the collaborators that have been participating in this project, and all you for your attention. Thank you.
Hello, everyone. I'm Gordon Arata from Children's Hospital, Los Angeles. And before I start, I would like to thank my collaborators on the study who are listed on this slide. I also want to thank Bionano for the opportunity to present at the symposium and describe our so far pretty modest experience with using optical genome mapping to detect rare genetic drivers in pediatric B-ALL. As we know, hematologic malignancies are common in children, and leukemia accounts for more than 25% of cancer diagnosis in young patients. And this audience does not need a reminder how genetic abnormalities are very important in risk stratification and treatment choices in pediatric patients with leukemia. And although in B-ALL, which is the topic of today's presentation, survival with current treatment reaches 90%, leukemia still remains a very significant cause of cancer death in children, and because of its high prevalence. So new trials are still there's still an effort to improve survival and improve outcome for children with B-ALL. And the focus is in particular on high-risk novel genetic subtypes of the disease, which have been characterized in recent years using advanced genomic studies. So these new genetic subtypes are very difficult to accurately diagnose and characterize by our traditional methods like karyotype and FISH. So there is a lot of effort in implementing new genetic testing methodologies for pediatric B-ALL. To address this need to detect novel genetic subtypes of B-ALL, at CHLA, we implemented a comprehensive genetic testing algorithm for all new diagnosed cases of B-ALL, which includes karyotype analysis and extensive FISH panel, but also includes routine use of chromosomal microarray testing and next-generation sequencing panel testing. We have our custom next-generation sequencing panel, which we call OncoKids, and which was developed for pediatric cancers. So even after implementing this comprehensive testing, and our recent retrospective study on 160 BLL cases has shown that the final genetic diagnosis remains unknown in 15% of our cases, which we still consider very high. So we are still actively looking into implementing further testing modalities, and we are in the process of validating RNA-seq for fusion detection and somatic exome sequencing, but we are still actively looking for other genomic testing modalities, including optical genome mapping. So we are all familiar with the recent multi-center study that showed some very exciting results with using optical genome mapping in characterization of leukemias. And namely, the study has shown that optical genome mapping can detect all copy number abnormalities and structural variants that were detected by karyotype at FISH, but it also detected additional changes missed by traditional methods in 11% of the cases, and then structural variants were refined in 24% of the cases. So based on this very exciting and promising data, we also decided to test optical genome mapping on our own cases and to do a small pilot study to determine if optical genome mapping can detect clinically significant genetic drivers missed by our comprehensive clinical and molecular evaluation. So it was a small study. It only included 8 patients, and the patient characteristics are shown on this slide, including some of the clinical details as well as results of previous genetic testing. But the thing that I really wanted to point out is that out of these 8 unknown cases, 6 were classified as high risk based on NCI classifications, EVA, showing that in these patients who remain unknown in terms of genetic drivers after our routine testing, there is enrichment for high-risk cases, and that stresses the importance of using additional methods to fully characterize these cases, because those are the difficult cases where genetic characterization and diagnosis will probably be very helpful in choosing better treatment. So to dive in directly into our optical genome mapping results, in 2 of these 8 cases, OGM allow us to detect previously reported and known driver fusions that have established clinical significance in BALL. And so I will show some details of these 2 cases. The first case was a 7-year-old and previously healthy boy who was diagnosed with standard risk BLL in 2018. However, unexpectedly at the end of the induction, he had residual disease and had to be reclassified into high risk and treated more aggressively. Initial genetic workup was negative for abnormalities that are known to be key drivers in BLL, but we did have some genetic findings that I wanted to show. So karyotype did detect translocation between chromosomes 2 and 5, as well as inversion of chromosome 14. And I have to mention, though, that at that time, we did not really realize what was the clinical significance of the translocation and of these 2 abnormalities. And chromosomal microarray did not show any gross copy number changes, but there was a focal deletion involving IKZF1, Icarus gene, which made us concerned that this might be a high-risk disease because Icarus deletions are associated with pH-positive and pH-like BLL, and they are by themselves a high-risk marker in BLL. So when we got optical genome mapping results for this case, it was very reassuring that the same abnormalities that we saw by cytogenetics were also detected by OGM. And so translocation 2, 5, and inversion 14 were also present by OGM. But more importantly, the optical genome mapping allowed to detect genes that were at the breakpoints of this 2-5 translocation and to fully understand its clinical significance. And so on chromosome 2, the breakpoint was within the gene SBTBN1, and on chromosome 5, within PDGFR beta gene. And so we immediately got excited about this finding because PDGFR beta fusions are associated with BCR-ABL-like B-ALL. And we went to confirm this finding by other methods. By the time we got OGM results, and we already had PDGFR beta FISH validated in the lab. It was not available at the time the patient was initially diagnosed. And so we were able to do FISH, and the FISH testing clearly showed that there was PDGFR beta rearrangement. So indeed, breakpoint on chromosome 5 was confirmed to be within PDGFR beta. We were also able to run the sample on RNA-seq. And RNA-seq also detected SBTBN1 PDGFR beta fusion as the top hit with pretty convincing supporting evidence. So why was this abnormality not obvious to us at the time of initial diagnosis? Although PDGFR beta fusions are quite well established, partner gene is very rare. It was only described, as far as I was able to find, in some rare cases of myeloproliferative neoplasms with eosinophilia, but this partner is either very, very rare or hasn't been found at all, and based on my research in PH-like BLL cases. And because of that, it was not incorporated into our OncoKids panel design. Also, we did not expect PH-like BLL subtype in patient who is otherwise standard risk, because those cases of PH-like are quite rare in standard risk BLL. So the patient would not have been even for this patient, FISH for PH-like abnormalities would not have been indicated. But it is still a very clinically significant finding for the patient, not only because PH-like BLL is associated with higher risk, but because PDGFR beta fusions are known to confer sensitivity to tyrosine kinase inhibitors. So the patient's leukemia would have been expected to be sensitive to imatinib, dasatinib, or other TKIs, and the patient would have been eligible for that type of treatment. The second case where previously reported abnormality was detected by OGM. It was a 3-year-old boy who was diagnosed again with standard risk BLL in 2018. He was initially treated in UK using standard protocols at the time for his type of disease. However, at the end of induction, he had residual blast, 2% blast, and he still had disease at 4% level in mid-consolidation. So he essentially had refractory disease, which was unexpected because he did not have any standard risks, any high-risk features at the time of his initial diagnosis. So he was referred for CAR-T treatment to CHOP, and that resulted in very brief remission. However, he relapsed again in 2019 with CD19 negative and CD22 positive BLL, so he was then referred to CHLA for another CAR-T trial. At the time we did genetic evaluation, his karyotype was normal, and CMA analysis was not done, unfortunately, but OncoKids panel did not show any known genetic drivers associated with a particular disease subtype. There was NRAS hotspot mutation present and some variants of potential and unknown significance, but nothing that would qualify as the key genetic drivers in BLL. So when we received OGM mapping results, to our surprise, although karyotype was found to be normal, and there was evidence of multiple structural variants, so evidence of really high genomic complexity, showing that normal karyotype was essentially a false negative. We probably were not able to get abnormal cells to grow in culture. And among all these different structural variants, what was particularly interesting and we think important was that there was a 3-way rearrangement that seemed to include chromosomes 1, 12, and 6. And at the breakpoints on chromosomes 1 and 12, there were important genes. On chromosome 1, it was MEF2D, and then on 12, it was FOXJ2. So the findings suggested the presence of MEF2D FOXJ2 fusion. Again, this was an exciting result because this is a fusion that has previously been reported. So we went to confirm this by RNA-seq, and again, the fusion was a top hit in our fusion by our fusion analysis tools. This is a finding of high clinical significance because MEF2D fusions are associated with a specific genetic subtypes of BLL. They have been described with a variety of different partner genes, but the gene that we saw in this case, FOXJ2, is exceedingly rare. I was able to find only one report which had one case of MEF2D fusion with this particular partner gene. But what is clinically significant about this finding is that MEF2D fusions are associated with high-risk disease and poor outcome, which would explain poor response to treatment and aggressive disease that this patient had. So this is just event-free survival and overall survival data, and from one of the studies dedicated to MEF2D rearranged BLL, and showing that these cases have inferior outcome than majority of other genetic subtypes of BLL, with the exception of BCR-ABL positive and BCR-ABL cases. In addition to known and previously reported fusions, we had findings of some novel candidate fusions that have not been described previously in BLL. And we had two cases with discovery of potentially new candidate fusions. The first case was a 16-year-old female diagnosed with high-risk BLL earlier this year. And she also had normal karyotype in our workup. FISH was positive only for low-level gain of PDGFR beta signal. And then she had some non-specific findings by CMA, but nothing that would classify it as important genomic driver. And the same was true for oncokids, which was negative. However, OGM testing showed evidence of multiple structural variants, again in a case that had normal karyotype, showing again how we often missed abnormalities by karyotype analysis. And most interestingly, there was evidence for a rearrangement between chromosomes 6 and X, with very interesting genes at the breakpoints, ATRX on X chromosome and BACH2 on chromosome 6. So OGM data suggested the presence of the BACH2 ATRX fusion. And so we were able to confirm this fusion by RNA-seq as well. And so we know by now that this is a real fusion, and we just have to find more evidence what is its functional significance and whether it is really an important driver for this patient's disease. However, both candidate genes are very important genes, and ATRX is an epigenetic regulator which has been shown to be mutated in some relapse cases of BLL, so it has a known association with BLL. And the BACH2 is a very interesting candidate because it is known to play a role in regulating B cell maturation. It has been described as a potential tumor suppressor gene in BLL because it is important to activate P53, and which is then required for elimination of pre-B cells that fail to productively undergo VDJ rearrangement. So both genes have some previous association with BLL, and the fusion has been confirmed as being real. And so our next step will be to look into its functional significance and potential to act as a driver, either by upregulating or possibly inactivating these partner genes. The second case with novel discovery was another high-risk BLL case. It was a 23-year-old male who was initially diagnosed in 2015. And at the time when we tested him and CHLA, he was undergoing third relapse after already having bone marrow transplant and CAR T therapy. Our testing showed normal karyotype and normal FISH. And chromosomal microarray had multiple findings, including Icarus deletion, again suggesting a high-risk disease and consistent with his history of high-risk disease. And oncokids testing was again negative. So in this case, again, OGM detected multiple structural variants, of which the most interesting was this inversion on chromosome 9, which had STRBP gene at one breakpoint and JAK2 at another breakpoint. And so for this fusion suggested by OGM, we still don't have confirmation whether the fusion is real, but we are excited about this result because JAK2 is obviously implicated in pathogenesis of PH-like ALL. In addition to detection of balanced structural variants, we know that OGM has ability to detect copy number abnormalities, and because we had copy number data from array testing for all of our pilot cases, we compared copy number abnormalities profile between optical mapping and chromosomal macroarrays, and we found very good concordance, which was really important that OGM to us that OGM can detect both types of abnormalities efficiently. And this is just one example of a case where we had nice concordance between the profiles detected by optical mapping and our arrays. The other thing that I wanted to highlight is that in our small cohort, we had 4 cases with completely normal karyotype in which OGM detected evidence of multiple structural variants. So again, showing that OGM has advantage over karyotype in its ability to detect abnormalities that are probably were probably missed by cytogenetics because the abnormal cells failed to grow in culture. And then another thing that was very important to me as a cytogeneticist was that optical mapping allows to see the chromosomal mechanism that leads to formation of abnormal gene fusions. Like an example of this MEF2D fusion, where we were able to appreciate that this was a complex rearrangement, 3-way rearrangement, which is consistent with these 2 genes being in such orientation on chromosomes that simple translocation would not have resulted in productive fusion. And then in the last case that I described, we see that the fusion most likely arose from an inversion on chromosome 9. And when we detect these fusions just by RNA-seq, we do not have any insight into chromosomal mechanism that leads to their formation. And I do believe that understanding the mechanism is an important component in us understanding these abnormalities, how they originate, and what are their consequences. So to summarize our results, among 8 cases that we tested so far, in 2 cases we detected known clinically significant and previously published and reported fusions, one associated with PH-like BLL, the other with MEF2D rearranged BLL. In 2 cases, we detected plausible candidate novel fusions. And then in all the cases except for 1, we detected structural variants which were not detected by our previously used testing methodologies. So in conclusions from our pilot study, we were able to see that optical genome mapping can detect both known clinically significant fusions as well as novel candidate fusions, showing both clinical and research potential of this technology. We saw very good concordance in copy number abnormality detection with CMA. We detected presence of structural variants in all of our cases that had normal karyotype. And OGM also can decipher chromosomal mechanisms that generate oncogenic fusions in contrast to RNA-seq. So with that, I would like to thank everybody who joined the presentation. I also want to thank all our faculty and staff at the Center for Personalized Medicine, all my wonderful colleagues, and special thanks to members of the Bionano Genomics team, especially Nick, Alex, Carl, and Sven. And I will leave you with this pre-COVID group picture of our team. For obvious reason, we do not have more current picture from this from last year, from 2020. But we are very optimistic we will have an updated new group picture sometime in 2021. So thank you again.
Here, I'm going to share our lab's experience with optical genome mapping in evaluation of genome-wide structural alterations that include both copy number changes as well as gene fusions in myelodysplastic syndromes, or MDS for short. I have no relevant conflicts of interest to disclose. So structural chromosomal variants are important for diagnosis and prognostication of MDS. And in some cases, detection of specific structural variants can alter therapy. Unlike somatic mutation profiling, high-throughput detection of structural variants has not been possible because of the presence of multiple repeat sequences in human genome. So right now, most well-characterized chromosomal alterations in MDS as well as AML are identified using a combination of traditional cytogenetic techniques. So these include conventional karyotyping, which has limited resolution because it's based on chromosomal banding patterns that is manually reviewed using light microscopy. FISH studies is more sensitive, but it has limited utility because it's a targeted approach. Chromosomal microarrays, they have a higher resolution and precision, but this assay is essentially ineffective in detecting balanced events such as inversions and translocations. Optical genome mapping, on the other hand, can potentially overcome this problem and provide a single platform for accurate identification of genome-wide abnormalities, in fact, all different types of structural variants at a fairly high resolution. So we decided to explore the utility of optical genome mapping in myelodysplastic syndrome. So why myelodysplastic syndrome? Now, and how important are structural variants in clinical management of myelodysplastic syndromes, or MDS? Now, based on the WHO classification guidelines, the diagnosis of MDS is dependent on recognizing dysplasia in at least 10% of cells in any of the three lineages. That would be granulocytic, erythroid, or megakaryocytic lineages. The morphologic assessment can be quite challenging, especially if the dysplasia is subtle or if the specimen is of limited quality. Now, on such diagnostically challenging cases, identification of specific cytogenetic abnormalities or structural variants is helpful to make the diagnosis of MDS. In fact, the structural variants that have been listed on this slide are considered as presumptive evidence of MDS in any patient with unexplained cytopenias. In some cases, the specific type of structural variant can guide treatment decisions. For example, the use of lenalidomide in MDS cases with isolated del(5q). Now, structural variants are very important in MDS for risk stratification. Now, despite identification of several gene mutations and molecular aberrations, the outcome of MDS is really and primarily dictated by the presence of larger chromosomal aberrations that are picked up by one of these techniques. Now, the treatment decision in myelodysplastic syndrome, whether it be to initiate the treatment or to change the therapy, it starts with risk stratification using the R-IPSS or revised IPSS scoring system. This includes blast percentage, degree of cytopenia, as well as the structural variants. And these include not only the number of structural variants, but also the type of alterations, and encompass both the copy number changes as well as translocations. At this time, the abnormalities for R-IPSS are detected. The R-IPSS uses only the abnormalities detected by conventional karyotyping. So in our lab, we chose to explore the utility of optical genome mapping with the following objectives. So the first is OGM able to detect known structural variants that we are able to pick up using traditional technologies? Second, are we able to uncover new aberrations that are clinically relevant in MDS? And thirdly, do these new aberrations alter the outcome in MDS in clinical trial setting? So we chose to explore the utility of OGM in two phases. So the first phase was our proof of principles pilot study, and the second phase is where we are testing several samples of MDS. We've tested nearly 50 samples so far, and it's an ongoing project. So first, for the proof of principles study, we tested 12 samples of MDS, 9 with cytogenetic abnormalities, and 4 were diploid karyotype, 4 samples of acute myeloid leukemia, and 2 samples of acute lymphoblastic leukemias. Compared to conventional karyotyping, we had a concordance of greater than 90%. Compared to the microarray testing, we had 100% concordance. In addition, the only reason for the lower concordance in karyotype was because of the inability of the OGM to pick up 2 small aberrations that were seen within a setting of composite karyotype, thereby not meeting the limit of detection criteria. Now, in addition to the abnormalities detected by these traditional technologies, OGM was able to identify additional aberrations that are of potential clinical significance. Now, for this proof of principle study, we used extremely stringent criteria to identify the clinical significance. One of these criteria included that the segmental alteration involved one or more of the myeloid genes that were implicated in any of the myeloid neoplasms. And of this cohort, all of the additional aberrations of potential clinical case importance were confirmed using alternate technologies, confirming the value of OGM. Since then, we have tested nearly 50 samples, and we have analyzed several of those, and we have confirmed the ability of OGM to detect all types of structural variants. And I want to highlight some of the important information that we're able to obtain from OGM using some examples. So here, the OGM is able to identify copy number gains and losses in autosomes as well as sex chromosomes with complete concordance with conventional cytogenetics and microarrays. Here we can see there is a large segmental deletion of chromosome 20 that the OGM is able to pick up that matched with the conventional karyotype. Now here is another example of MDS where we are able to identify deletions in chromosome 5 shown as in red. We are able to identify precise genomic details of the deletion as well as in addition a gain of chromosome 6 that is apparent. This was not picked up by conventional karyotype. In addition, as we noticed, the conventional karyotype shows deletion of chromosome Y. So when we look into it, although the circus plot, the deletion of chromosome Y is not that apparent, we are able to appreciate that in a copy number plot, where the CNV of chromosome Y is less than 1 compared to a random control sample from an unrelated male. Now here we are able to see that the OGM is able to identify translocations between 2 different chromosomes, in this case chromosome 2 and chromosome 20. And at the same time, we were able to map out using OGM a complex chromosomal event. Here it's a derivative chromosome that involves segments from chromosomes 1, 12, and 5. Now, using karyotype, this level of complexity involving this derivative chromosome was not apparent. Now, OGM was also able to show the breakpoint heterogeneity in genomic aberrations that is not apparent by conventional techniques. Now, this, as we can see here, there are 2 different sizes for the chromosome 20 deletions, and we can also appreciate the 2 different fusion products here that translate to the deletions with of different sizes. Now, this is important for us to pick up because the clonal heterogeneity is responsible often for the resistance and relapses in hematologic malignancies. Now, the OGM is also able to clearly detect and map out the inversions, and here it's able to map it out at the gene level. This was an example of MDS with inversion of chromosome 3 that involves the MECOM or the EDI rearrangement, which is considered as a poor prognosis. Now, this level of gene detail essentially nullifies the need for an additional FISH assay to confirm the involvement by MECOM. Now, similarly, this is another MDS case which had a translocation 911 that was detected by conventional karyotyping. By OGM, in addition, we were able to identify a large deletion involving chromosome 11, and that encompassed the MLL or KMT2A gene. And this we were able to confirm by FISH and map-back FISH as well. Now, more importantly, OGM is able to uncover submicroscopic or cryptic aberrations that are clinically relevant. And these are often cryptic by other standard of care technologies. So this is a case of MDS, where we were able to detect a TET2 deletion involving chromosome 4. This was cryptic by karyotype because it's generally very small, and the labs do not routinely perform FISH for this specific deletion. Interestingly, this case by next generation sequencing studies also had a mutation in the SF3B1 and an additional frameshift mutation in TET2, thereby confirming the biallelic inactivation of TET2. The point of this is essentially that all of the aberrations, except of course the mutation data, were obtainable using one platform or technique, avoiding the need for multiple confirmations. Now, this is also important because we are aware that the biallelic TET2 alterations are pretty frequent in myeloid neoplasms, and the deletions of TET2 are essentially missed if we do not perform a high throughput assay for structure variant detection. Now, to summarize, optical genome mapping essentially provides a single platform for accurately identifying all different types of structure variants, thereby avoiding the need for multiple confirmatory assays. Now, this makes it faster and less expensive overall. And in summary, I believe that this technology has immense potential to serve as a high throughput platform for structure variant analysis in the clinical laboratories. And I wanted to end the talk by acknowledging everyone that's responsible for implementing OGM in our lab. That includes Dr. Guillermo Garcia Monero, as well as Hui Yang, who's been hard at work on the bench, as well as my chair, Dr. Medeiros, and Dr. Lutra, who's leading the molecular diagnostics lab. Thank you.
Hi, I'm Valentin Lestringant, cytogeneticist at the Hematology Laboratory of CHU Amiens-Picardie, and I will present to you our work on the use of Bionano Genome optical mapping in a series of ALL cases. To begin, we choose to focus on AL because of the frequency and structural diversity of cytogenetic anomalies in these pathologies. The characterization of these abnormalities remains a central point in the management of patients with ALL by their prognostic and therapeutic impact. This diversity requires the use of several conventional and molecular cytogenetic techniques to establish the most exhaustive genetic profile. And so we investigated if optical mapping had the capacity to replace all standard techniques. Our objectives were to evaluate both analytical performance of the optical mapping technology and the computer tools for data processing and filtering. In summary, are operators able to easily isolate anomalies of interest from other signals? We studied 10 AL previously genetically typed that were selected to obtain a representative panel of major prognostic impact anomalies and their structural diversity. Optical mapping data were interpreted independently by blind operators before comparison. And secondly, we investigated the possibility of detecting copy-neutral loss of heterozygosity. Selected cases were 10 B or T ALL treated in Amiens. Reference techniques were performed in Amiens and Rouen for karyotype and FISH, and in Lille for SNP array and RT-MLPA. Optical genome mapping was performed using bone marrow DMSO pellets and the platform Gentian in Clermont-Ferrand. The rare variant pipeline was used for the analysis, and blind operators were Joyce Lee, Bionano engineer, and myself. About the selection of anomalies, in SNP, we retained all anomalies with a size superior of 1 MB and or involving a list of 186 genes of interest involved in leukemogenesis, and with a minimum size of 20 kb. For other techniques, all the anomalies found were selected. And for optical mapping, the anomalies were first extracted according to the size and confidence score criteria, and then selected using similar criteria as in SNP. About patient characteristics, we had 6 B-ALL and 40 ALL, 7 children and 3 adults. Using standard techniques, we found a total of 80 anomalies meeting inclusion criteria, and we excluded 2 CNLOH that were separately analyzed. Using optical mapping, after extraction, we obtained a total of 948 calls. After filtering and analyzing these calls, 89 anomalies for the first operator and 84 for the second were retained. In total, 73 of the 80 referenced anomalies were found by optical mapping. 7 anomalies were missed, and 12 anomalies not described by standard techniques were newly found by optical mapping. Here are the anomalies correctly found in optical mapping. As you can see, the majority of anomalies of clinical interest were found, including, as examples, hyperdiploidy, 21 intrachromosomal amplification, and some recurrent translocations. Here are the anomalies that were missed by optical mapping for reasons that I will detail. In the first case, trisomy 3 and trisomy 4 were missed, but clearly visible in the raw data using the whole genome CMB representation. The 3Q region was more gained, which allowed it to exit filters for data extraction. In the same way, 22 gain pretty visible in raw data was missed in another case. Subclonal work chromosome gain 22 was missed too. It was found in 7 of 22 metaphases in karyotype, but quantified at 7% of nuclei by fish. So this is below the detection limit of optical mapping and suggests an in vitro proliferative advantage of this subclone. Here is the case of a near tetraploid subclone that was found in 6 of 19 metaphases, where close to tetraploidy with a variable number of chromosomes, but with a low resolution at the limit of the classifiable. It was neither found by SNP array, so that may suggest a true tetraploid subclone. 2 anomalies were missed because involving a pseudo-autosomal region. This includes a deletion leading to P2LY8 CRLF2 fusion. Pseudo-autosomal regions are, as other regions with highly repeated patterns, centromeres, telomeres, are indeed not easily accessible using optical mapping, as well as other techniques. To finish, To finish, this VPRB1 deletion was correctly found by the technology, but missed by operators during the analysis, as it was hidden by recurring deletions of the IGL locus. Here we are showing anomalies newly found in optical mapping. 3 of them concern rearrangements well described in ALL, leading to LMO2 TRA fusion, TRB MIC fusion, and IGH CEBPB fusion. This translocation involved various breakpoints and led to partner overexpression, so no transcript could be found using MLPA. However, FISH are non-systematically performed on these targets, so we decide to confirm them using FISH probes. Now, as an example, the case of the LMO2 TRA rearrangement. This case was actually more complex because involving rearrangement of TLX1 as well. Here you can see the different derivatives, the blue maps from rearrangements of TLX1 with TRA at the top and LMO2 with TRA at the bottom. These derivatives align with the reference maps in green, with chromosome 14 at the TRA locus at the bottom, and at the top with chromosome 10 and TLX1, or chromosome 11 and LMO2. Note that crossed lines in LMO2 derivatives symbolize an inversion. As shown by the pink dotted lines, 2 breakpoints are present in the TRA locus. The first one in the middle of the gene was shared by the 2 derivatives of the middle, and the second on the right was shared by the 2 other derivatives. So if we schematize chromosomes, we have a total of 4 breakpoints, 1 in the chromosome 10 involving TLX1, 2 in the TRA locus on chromosome 14, and another one in the chromosome 11 involving LMO2. Interchromosomal translocations then resulted in 2 derivatives from a pretty classical 1014 TLX1 TRA translocation that was found by karyotype. And the TRA fragment between the 2 breakpoints will be inverted and inserted into LMO2 onto the 2 chromosome 11, resulting in a LMO2 TRA rearrangement. Here we show the FISH analysis using the LMO2 probe that shows no split signal, and the TRAD probe showing a typical split signal corresponding to the TLX1 translocation and an extra green signal compatible with the insertion into LMO2. Regarding other newly found anomalies, we have 2 other translocations to control, 2 high complexity rearrangements, and there remain 6 deletions, 2 gains, and 1 duplication. 4 of them involved no gene of interest but were higher than 1 MB and only found by the CNV tool. Turned out to be false positive signals. The remaining anomalies were confirmed by the SNP array raw data. CMLOH detection, which is not possible by optical mapping in theory, was investigated using the de novo assembly pipeline. We removed all filters, including database variants, to show all variants detected by optical mapping. Then we looked into how much variants were called homozygous by the SV tool, in the LOH regions previously detected by SNP. As you can see, in the two samples who carried CNLOH, the rate of homozygous variants was above 90%, which was pretty much higher than the rates in these regions for patients free of CNLOH or deletion. To finish, it's important to mention that we faced a problem of cross-sample contamination. 17 anomalies were in fact exactly the same between sample 4 and sample 7, with very low molecule counts for the contaminated sample, which is sample 7, and including a PCR Belson translocation. And this makes us consider the implementation of system to identify DNA molecules, such as a code boring system. To conclude, we found the Bionano optical mapping technology to be pretty mature. It provides a resolution and analytical sensibility as expected and promised. Remaining defaults are anomalies occurring in pseudo-autosomal region, centromeres and telomeres, as well as gains and losses of chromosomes in small fraction. Cross-sample contamination also remains a problem to be solved. General use in diagnosis obviously needs larger cohort trials and needs to be tested in other hemopathies, but overall optical mapping is already a promising tool for research of new variants and for the resolution of high complexity rearrangements. Thanks to all the people who contributed to this work, and thank you for your attention.
Hello everyone. My name is Bo Chen, representing Beijing Lu Daopei Institute of Hematology, Lu Daopei Medical Group. I'm here to share with you the preliminary study using Bionano's optical genome mapping technology to decipher genomic structural variations in acute leukemia. This project is in cooperation with Genomics. Genomic structural variation include fusion genes and gene copy number variations. More than 100 fusion genes and dozens of gene copy number variations have been recognized in the guideline for leukemia published by WHO in 2017. They have essential pathological and clinical significance, but there are dilemmas in clinical applications as well. The distribution of fusion in acute leukemia is typical a long-tail phenomenon. In our recently completed RNA-seq project for 1,000 acute leukemia patients, here at Daopei Hospital, a total of 686 driver fusion events were detected, of which there were 227 distinct fusions. A considerable number of fusions in the tail position were only detected in one patient, but their identification is critical. For example, the PDGFR beta fusion variations share common features that are responsive to tyrosine kinase inhibitor or TKI targeted therapy. For fusion genes in which both partners are known and fixed, for example, BCR ABL1 fusion, detections can be performed using a set of PCR primers. On the other hand, some fusion genes may have multiple partners. Their detection is very challenging. Gene copy number variation, CNVS, also matter in leukemia, but methodology for detecting CNVS are more problematic. The Bionano optical genome mapping technology is unique and can comprehensively analyze genome structure variations with high resolution. We intend to evaluate its performance in analyzing the structural variations in the leukemia genome. For this study, we have completed Bionano analysis for 14 cases of leukemia, which includes 5 AML, 8 BALL, and 1 TALL patients. The blast cells in the sample ranged from 37 to 99%. Methods used in the evaluation in comparison with Bionano include karyotyping, multiplex PCR, which can detect 41 fusion genes developed by Dalbei Hospital, and high throughput messenger RNA sequencing. For fusion genes, we compared Bionano, karyotyping, and RNA-seq results. For CNV, we focused on a list of 46 genes reported to be CNV drivers in leukemia. We used RVP mode for structural variation analysis in Bionano and only count the highly credible variations. Expression of SV-associated genes were analyzed against the 300 leukemia driver genes, which were summarized by Dalbei Hospital, and we believe they are significant to leukemias. Data obtained by Bionano ranged from 82 to 657 gigabytes, and mapping rates ranged from 72 to 87%. A total of 10 cases were detected positive for pathologically significant fusions. In comparison, RNA-seq confirmed all 10 fusions. PCR screening missed 4, including the rare PAX5, EBF1, and recently reported MEF2D EP300 fusions. 5 fusions, including the above 4, were missed by karyotyping, and also missed was the well-known KMT2A fusion. Bionano detected a non-reciprocal translocation corresponding to this fusion, but none other methods could. In the following slides, we report the significant findings in some of the cases detected by Bionano. In case M1, both karyotyping and Bionano revealed an inversion in chromosome 16. Karyotype reported a trisomy 22, but Bionano analysis only showed amplification of the long arm of this chromosome. More detailed analysis indicated that karyotyping may have mistakenly reported the trisomy. Bionano further revealed a 500 kilo BP and a 400 kilo BP deletion in the inversion. RNA-seq confirmed the CBFB MYH11 fusion transcription caused by the inversion. In case B1, Bionano revealed translocations of PAX5 and SD1 and CRLF2 at IGH and corresponding locations. We can identify there were irregularities of chromosome 5, 9, and 14 in karyotyping, but it failed to determine both translocations. Fusions related to PAX5 and NSD1 have been reported in acute leukemia, but fusions of these two genes together has not been reported before. this is the first time. In the novel PAX5 and SD1 fusion, the breakpoints of both genes were the same for their canonical locations. So we believe the fusion is also significant. It has been reported CRLF2 at IGH leads to CRLF2 overexpression, especially in BCR-ABL1-like BALL cases, which is typically associated with poor prognosis. This case, CRLF2 expression was highly overexpressed. In fact, it was the second highest among 393 BALL cases in our another study. So the translocation detected by Bionano is helpful. In case B2, which reported as normal in karyotype, Bionano revealed an 8.6 million BP deletion in chromosome 5 and EBF1 PDG-FR beta fusion. RNA-seq confirmed the fusion transcript and PDG-FRB beta overexpression due to the fusion, which is responsive for TKI targeted therapy, and so has clinical significance. The karyotype of case B4 revealed multiple subclonal translocation events as a major monosomy 9. Bionano revealed a MEF2D BCL9 fusion due to a microdeletion in chromosome 1, which could not be detected by karyotyping. Bionano CNV analysis reported a CDKN2A deletion, which could be a consequence of the monosomy 9. Bionano analysis also showed at least 2 amplification events in chromosome 1. Furthermore, a reversal of direction of the BCL9 is revealed, which causes to form functional MEF2D BCL9 fusion, in addition to direct breaks. RNA-seq confirmed the fusion transcription. This fusion leads to BCL9 overexpression, which is also associated with poor prognosis in clinical application. Case B5 was reported with complex karyotype aberrations and multiple translocation events. However, 2 significant events, an EP300 fusion associated translocation and a translocation between chromosome 19 and 21 were reported by Bionano, but totally missed by karyotyping. Bionano analysis also indicated an inversion of a reverse EP300 between a CNF384 gene and its partially duplicated copy. ERG and EPOR were neighbor genes of chromosome 19 and 21 translocation, as revealed in Bionano analysis. RNA-seq data showed that both genes were overexpressed in this case. It is known that EPOR overexpression is the driver gene for certain BCR-ABL1-like BALL cases and is responsive for TKI targeted therapy as well. ERG overexpression is listed in the WHO guideline as a poor prognosis marker in acute leukemia. There was no fusion identified in case B8 by any of the analysis. However, Bionano did reveal 11 kilo BP microdeletion in chromosome 13, but none other analysis did. This microdeletion occurs at immediate upstream of the FLT3 coding region and involves the PAN3 locus. A recently published paper reported microdeletion in this region is a recurring event in BALL. This microdeletion results in increased cis interaction between the FLT3 promoter and another hydroxyl enhancer located in distance to the breakpoint, with subsequent upregulation of FLT3 expression. RNA-seq confirmed relative overexpression of FLT3 and impaired expression of PAN3 in this case. In summary, our preliminary data support that the bio-optical genome mapping technology is a powerful tool for deciphering the genome structure variation. It can perform genome-wide SBS analysis in one test with high throughput, high resolution, and good accuracy. It also detected a novel PAX5 NSD1 fusion for the first time, a microdeletion associated with FLT3 dysregulation and other structural variations associated with the aberrant gene expression. Moreover, the numbers of SBS reported by Bionano is more than 10 times of those whose pathological significance is interpretable, and provides more SV evidence that requires further study. The authors of this study would like to thank folks from Grand Omnics and colleagues from Dalby Hospital for their work and collaboration. Thank you for your attention. Happy New Year and best wishes for 2021.
Welcome and thank you for listening to my talk. I'm Barbara and I'm supervising the routine diagnostic lab of genetics of hematological malignancies at the University Hospitals of Leuven in Belgium. And I'm very pleased to present you our recent work on optical genome mapping. And this new technology really works really well. And so we are currently implementing this technique for the routine genetic diagnosis in acute lymphoblastic leukemia. So in our routine lab, we perform many different cytogenetic and molecular genetic testing procedures like karyotyping, FISH, multiplex ligation-dependent probe amplification, different kinds of PCRs, and of course NGS. And using all these different techniques, we obtain a panel of genetic results, which are all needed, in fact, as a clinical tool to help with the diagnosis and prognosis of hematological malignancies. Moreover, some of these results are needed to determine the best treatment strategy for the patient. On this slide here, I summarize the WHO classification of the hematological malignancies. So broadly, these can be divided into myeloid and lymphoid neoplasms, and based on morphology, immunophenotype, clinical and genetic features, they can be further classified into different subcategories, as you can see here. And for today's talk, I will focus on the precursor lymphoid neoplasms. And broadly, you can classify this group of diseases into B and T lineage lymphoblastic leukemia and lymphoma. Some of the B-ALLs can be further subcategorized according to some recurrent genetic abnormalities. So what is ALL? Most patients present with the consequences of bone marrow failure. And ALL is diagnosed by blood and bone marrow examinations, including genetics. ALL is characterized by the accumulation of malignant immature lymphoid cells in the bone marrow. It is the most common malignancy in children, representing almost 25% of pediatric cancers. The outcome is good, especially in children, but urgent medical intervention is required to prevent inferior outcome. So current treatment protocols in ALL require extensive genetic testing. And here in this table, I summarized all the aberrations with clinical significance in terms of risk in ALL. As you can see, these are different types of aberrations, including recurrent structural rearrangements, whole chromosome copy number aberrations, and also submicroscopic deletions. All these genetic results are needed fast in order to be able to stratify the patients into different treatment arms. So currently, in fact, an extensive panel of tests is needed to be able to obtain all these necessary results. So you need a classical karyotype, you need up to 10 different FISH experiments, and moreover, you need MLPA or SNP arrays to be able to pick up the submicroscopic aberrations, and you need some PCRs, for example, to be able to pick up the Stiltal-1 rearrangement. Performing all these different techniques is costly and laborious. And most labs, they use cascade testing, which results in long turnaround times. So I guess you can imagine that we were very interested to test this new technique called optical genome mapping, which claims to be able to detect all these numerical and structural abnormalities in just one assay. So using this technique could really reduce costs and turnaround time and might even increase diagnostic yield. In order to validate whether optical genome mapping could replace our current analytical strategies in the routine lab, we analyzed 10B and 10T ALL cases that were previously analyzed with our current test panel. 3 different bioinformatics pipelines were used. So we used the de novo assembly pipeline, which first constructs, in fact, a de novo assembly and then compares the resulting assembly with a reference. This pipeline is highly sensitive in detecting structural aberrations in a diploid genome, but this approach may miss structural aberrations at very low variant allele frequencies. So we also use the rare variant pipeline, which is specifically designed for the sensitive detection of somatic structural variants occurring at this low VAF. Also, a copy number analysis pipeline is embedded in the other 2 pipelines, and we also use this one. So we also filtered our data using our own custom filters. So all copy number aberrations longer than 5 megabase were reported, and submicroscopic aberrations smaller than 5 megabase were only reported when a clinically relevant locus is involved. In the next couple of slides, I will illustrate the features and the possibilities of optical genome mapping by showing you examples of 2T and 5B ALL cases. The first case is a 6-year-old child presenting with cytopenia and hyperleukocytosis. Unfortunately, the standard karyotype failed for this TALL case. This is one of the disadvantages of karyotyping because you need cells that grow in culture, which occasionally might fail. So we thought that this would be a good sample to check if optical genome mapping would be able to pick up some aberrations. So the output of optical genome mapping is a circus plot, as you can see here on the slide. And the outer circle depicts the classical cytogenetic bands. Here you can see the structural aberrations which are indicated by colored dots, and the inner circle shows the copy number aberration profile. And so you see 2 copies of all the autosomes and 1 copy of X and Y. So at a glance, in fact, you can see that using optical genome mapping, we could detect here a translocation between chromosome 5 and 11, resulting in a fusion of TCF7 and SPI1. And detecting this is very important because this translocation was previously reported to be associated with very poor survival in ALL. We also detected a submicroscopic deletion in chromosome 9p, leading to loss of CDKN2A. And interestingly, in fact, this case was also analyzed a few years ago in research, and the same results were also detected by single-cell sequencing by a group of the KU Leuven, and it was reported in 2018. If we zoom in then to see the genome browser view, you get a consensus map that nicely supports this translocation, and it also illustrates the mapping of the breakpoints to the genes that are involved. The second TALL case we included because it had a translocation 1114 into 3 out of the 15 mitoses that we analyzed, and this case also had a cytogenetically cryptic Stiltal1 rearrangement. So we were wondering if Bionano would be able to detect both of these aberrations. And at first we detected only the 1114 translocation, but I decided to check this region in chromosome 1 in detail manually, and I could see in fact that there was something going on with the labeling pattern. And after resetting our filter parameters, we could also nicely detect this submicroscopic deletion on chromosome 1 that leads to the Stiltal1 rearrangement. Again, if we zoom in, you get a consensus map which shows you the location of the breakpoints. The third example is a BALL case with hyperdiploidy. So the karyotype, as you can see here, shows a high hyperdiploid karyotype, so gain of a lot of chromosomes, which is prognostically favorable. And we were wondering if optical genome mapping would be able to detect this correctly. So, and here you see the results. So genome imaging nicely detects all the chromosome gains and gives in fact the same copy numbers as found with classical karyotyping. So 3 copies for chromosome 4 and 4 copies of chromosome 21. The software also provides a whole genome copy number view. This is a visualization similar to the one of CGH array data, which might be nice to use. The next case is again a BALL case for which the karyotype failed because the sample arrived after a few days and the cells didn't want to grow anymore. So we were wondering if Bionano would be able to rescue and to provide us with accurate results. As you can observe on the circus plot, optical genome mapping identified hyperdiploidy in this case. But to our surprise, FISH analysis showed monoallelic losses. So FISH in fact suggested a low hypodiploid clone. And this low hypodiploidy was also confirmed by CGH array. So if we now go back to one of the first slides, you can see that hyperdiploidy is associated with a good risk, while low hypodiploidy is associated with high risk. So it is very important to get diploidy right in ALL. So we have been trying to find solutions to be able to correct diploidy. So I plotted the zygosity of the variants, chromosome per chromosome, and you can see that for the chromosomes of which we know they have only one copy in the tumor cells, you get almost no heterozygous variants, which would suggest loss of one copy of this chromosome in the tumor cells. So if you use this to correct the baseline, you get the same low hypodiploid karyotype as we found with RACGH. So of course, this kind of manual approach is only possible in cases where you have a lot of tumor cells in the sample. So we are currently working together with Bionano Genomics in order to optimize their pipelines based on this approach. The fifth sample that we tried on the Bionano, I included because it is a very complex case with multiple aberrations as shown here. And this case also has multiple clones. And this case also had fewer blasts in the bone marrow than the previous cases we analyzed. And using FISH, in this case, we identified an intrachromosomal amplification of chromosome 21. So here you see the Bionano results, and almost all the aberrations detected in the karyotype are also seen here. So you see the abnormal chromosome 3, the deletion in chromosome 4, etc. Except for the gain of chromosome X, which is here present in 2 out of 12 mitosis. This is not detected using optical genome mapping. And I think we are here at the detection limit of the technique. So the company reports it to be at about 5% FAF. And if you do the calculation, well, the plus X is at 8% FAF. So this could be around the detection limit. And for example, this ADD 10P11, which is also found in 2 out of 12 mitosis, is nicely picked up by the technique. So I think we are really at the border here. This case also illustrates, though, that optical genome mapping is not able to identify multiple clones. So using karyotyping, we found 2 clones in this case, so one with 47 and one with 45 chromosomes. And yeah, this is in fact a known limitation of DNA-based techniques, that you cannot detect multiple clones. The submicroscopic deletions, though, like this deletion involving several exons of the RB1 gene, are nicely detected, and these results are concordant with MLPA results. And which is also very nice is that the intrachromosomal amplification of chromosome 21 is picked up, and the number of copies of the RUNX1 gene involved in this aberration is exactly the same by FISH and by optical genome mapping. So both report over 8 copies of RUNX1. Another nice feature is that Bionano also allows you to zoom in and to get a circus plot of the chromosome of interest. And so because of this, you can see nicely here the details of the chromotripsis, which leads to this intrachromosomal amplification of chromosome 21. The 6th case I included because I think it really nicely shows the power of this technology. So it's also a PALL for which the karyotype shows an extra chromosome X, an abnormal chromosome 1 and 5. There were doubts also about chromosome 7, and we found an extra chromosome 21. And optical genome mapping immediately at a glance, in fact, shows you that the abnormal chromosome 1 and 5 result from a translocation 1-5, resulting in this the fusion of these two genes. And this is nice and very important because chromosomes from ALL cases can typically be very ugly and difficult to karyotype, and it's very important to detect this kind of translocations, because this one immediately classifies this case as a Phi-like ALL, which is associated with very high risk. So we could immediately correct the breakpoints of the karyotype, and we could also see that the doubts about chromosome 7 were not necessary, because here we can see that chromosome 7 is normal. In addition, you can again see here that optical genome mapping is able to pick up all the key submicroscopic copy number aberrations, like the small deletion resulting in the deletion of the IKZF1 gene. This I also wanted to mention. So, which is important for this patient is because of this translocation we detected. This was previously reported to encode a constitutively active tyrosine kinases. So this patient with a bad prognosis could benefit from treatment with a tyrosine kinase inhibitor like imatinib. The last case I included is a BALL with a rather simple karyotype. So this karyotype shows the derivative 18 resulting from a translocation 119, which is an aberration associated with intermediate risk. And you will immediately see why I included this one. So the de novo assembly shows the extra material on chromosome 1, resulting from the derivative 19. And the rare variant pipeline shows the same gain. But if we zoom in, the rare variant pipeline shows a much more complex pattern. So you can nicely appreciate that there is an inverted insertion of a 140 kb segment of chromosome 19p into chromosome 1q here. So these two chromosomes, one who look normal, well, now we know that at least one of them is not. And we could confirm this result using FISH. So FISH was performed on a bone marrow aspirate after culture, and we investigated 10 metaphases. And in 6 out of 10 metaphases, we found 2 fusion signals. So one on a derivative 19, and the other one on a chromosome that looks like a normal chromosome 1, but has a cryptic insertion of TCF3. So I can conclude that we had very nice result with optical genome mapping in acute lymphoblastic leukemia. So for all the cases we analyzed, an informative result was provided. Also, all submicroscopic aberrations were found. So we had an excellent concordance with MLPA and no false positives. Also, all recurrent translocations identified by the classical test panel were correctly identified. Again, no false positives. We also had an excellent concordance of whole and partial chromosome gains and losses. But of course, our study also highlighted a few limitations. So ploidy and multiple clones are not always correctly determined. Moreover, we also found extra aberrations in cases with failed or normal karyotype, and in some cases we could even correct the karyotype or refine the breakpoints. We also found some extra aberrations, including rearrangement of the TCR and the IGH genes. So to conclude, optical genome mapping is a very promising technique to identify structural and numerical aberrations in ALL, and this within 1 week from sample collection. So, what will change in our lab for ALL at diagnosis? So at this moment, we perform these 4 techniques. MLPA will disappear and will be replaced by optical genome mapping. Also, the number of FISH experiments per ALL case will drop to 0 or 1 test per patient. and also the number of PCRs will drop. At this moment, we are still planning to keep on karyotyping, but because we will already have the result of the optical genome mapping, the time that we put into the karyotyping will also significantly reduce. In future, we are planning to further optimize optical genome mapping, mainly for the determination of the ploidy. And we also plan to validate and implement this new process for other hematological malignancies in the near future. And with that, I will end my presentation, but not without thanking all my colleagues for their help and support. First of all the people from my routine lab. And I would also like to thank the people from other labs and departments in the University Hospitals Leuven and in the University of Leuven who were involved in this study. And thank you for listening.
Well, good afternoon, everybody. It is indeed my pleasure to be able to present the results of our recent multi-center trial that we did evaluating optical genome mapping in patients with acute myeloid leukemia. I want to thank the Bionano folks for the opportunity to present this exciting data. And I usually start off introducing the topic and showing some really nice animations. But after 2 days of having this webinar series, I'm sure you are very well versed in optical mapping. So I'm going to jump right into the whole study and the results that we found. Okay, so if we really look back at karyotyping and its association with cancer cytogenetics, it's important to realize that we've known about this association for well over 100 years, going back to Boveri, who first observed abnormal chromosomes in solid tumors. Over the course of the last 100 or so years, cancer cytogenetics has indeed become an integral part of the workup of patients with cancer. And we do know that we need to target both structural abnormalities as well as copy number abnormalities. We've been doing that by routine karyotyping, and more recently, I guess since the late 1980s, using multiple rounds of FISH with different types of FISH probes. And more recently, with the introduction of chromosomal microarray, we've been able to assess the whole genome and look at far smaller aberrations, submicroscopic aberrations, which go well beyond the visual aspect of karyotyping, but cannot detect balanced structural rearrangements or even map the chromosomal changes that we see and tell us exactly where they are coming from. And so a single technology that covers both structural abnormalities and copy number changes is highly desirable. And when I first learned about optical mapping, I guess myself and anyone else that looked at the technologies immediately thought, well, this potentially could be a great fit to achieve this goal. And I got a long way to achieving this goal with the creation of what I call the Cancer Genomics Research Network, and this was established for rapid assessment of emerging technologies. There are clear benefits of a research network. It enables us to really get hold of rare samples. If we think about these rare samples at an individual institution, the likelihood of getting them in a timely manner for a study is very small, but as we leverage our network, we can get access to these rare samples in a very expeditious manner. And it enables us, in addition to leverage a diversity of samples across multiple sites, many additional advantages like spreading the costs. And so there are many potential uses for this, including rapid assay validation and then assessment of clinical utility, which those of us working at large academic medical centers will need to enlist the collaboration of our local clinicians. And just to let you know, in the United States, that for the current first example of this type of network, these are my colleagues who were just fantastic in this collaboration. And here you see that's Alan Lennon, Jim Broach, Linda Bourne, Min Fang, Rashmi, Kanadal Shamana, Ravi Kola, and Yasmin Akari. The culmination of the multi-center study did come with our pre-publication in medRxiv, which you can see the listing over here. For those of you interested in actually delving a lot deeper into the full results, I'm going to concentrate today really on some of the highlights of this study. The aims of the study were relatively simple. We wanted to look and establish the clinical diagnostic capabilities of optical mapping in cancer patients with AML. We did a blinded assessment and comparison of the results generated by optical genome mapping to results generated from routine clinical testing that was achieved in a clear laboratory setting using current gold standard cytogenomic techniques. We had 100 patients where we received either peripheral blood or bone marrow samples, and these were patients who were subsequently were initially diagnosed and subsequently treated using intensive chemotherapy. The clinical samples were initially received and processed at 8 separate CLIA/CAP certified labs, and those are my collaborators that I showed you on the previous slide. And actually I was going to say almost all of them, but in fact all of them, 100% of them had karyotype, which is the gold standard. You'll see that only about 19% of cases had FISH, and only a very small fraction of them had chromosomal microarray. And again, this is routine clinical testing ordered by our clinical heme-onc clinicians. 80% of the samples in the study were unselective consecutive samples. We really wanted to get an idea in a consecutive manner how optical mapping would perform. But then we did want to really test the system by enriching for a wide range of anomalies. And these anomalies were chosen by my colleagues at their individual places. And when the optical mapping was done, those results were in fact blinded. The study was carried out under IRB protocols that were approved at each institution. The primary hypothesis was that optical genome mapping will provide clinical diagnostic results in a single assay, concordant with those achieved using multiple gold standard technologies. The secondary hypothesis was that optical mapping will uncover novel cytogenomic aberrations that will provide further insight into the pathogenicity of certain cancers. And certainly this is future work that, the results of the study lays the groundwork for multiple future studies. We assessed the clinical utility of optical mapping in 2 primary ways. So one, and the most basic of ways, was literally just to see the concordance. What we saw by routine testing, do we see the exact same thing by optical mapping? And that included both structural variation as well as copy number changes. And all of these were diagnosed in our CLIA/CAP approved laboratories. And then the second aspect was to say, over and above picking up what we saw by routine testing, were the things that we could pick up using optical mapping that were not identified by routine testing. From many of the studies that have come out from Bionano, it is apparent that there would be many additional findings. And so knowing that in advance, what we decided is that with all the additional findings that we would get across the 100 specimens, and indeed there were many, many additional findings, we wanted to focus specifically on ones that we could hang our hat on as potentially being clinically relevant. And the way we did that was by filtering all the additional optical genome findings with AML-specific genes. So we made comprehensive gene lists that we had across the 8 different clinical institutions, as well as the common AML-specific probe regions. And so we use these as a filtering, as a filter to further bring out specific optical mapping findings that crossed over with these regions. So we further assessed them using the following criteria. We looked at its presence or absence in the Bionano database, so that was a very large help because we wanted to rule out just common variants. And then we also only included sizes that were greater than 5 kb for insertions or deletions, as well as greater than 5 megabases for CNVs. And remember, these criteria were what we further added for the additional findings that optical genome mapping did over and above the routine methods. We decided that once we got the final filtering with those criteria, we would use a expert curation panel, and we dug deep into, we didn't have to dig deep, we actually utilized the expertise of our colleagues that were on the study, who got together to discuss the clinical significance of additional structural variants that were identified by optical mapping after we used all those filters. And then the final list of additional structural variants and copy number changes was compiled based on 2 tiers of clinical significance. So identification of 1 or more aberrations which are used in the ELN 2017 risk stratification system, or those that change the ELN risk status. And secondly, identification of abnormalities that may or may not be specified in the ELN 2017 risk stratification system, but for which sufficient published data demonstrates clinical significance. It was very important that we confirm any clinically significant findings, and we certainly went about and did that, and we confirmed every single one of them either by microarray or a PCR or Sanger approach. And let's get into the results. We stratified the cytogenomic results that were reported into 5 categories. So negative karyotype, either aneuploidy or partial aneuploidy, rearrangements such as translocations and or inversions with aneuploidy, rearrangements without aneuploidy, and then complex abnormalities with greater than 3 abnormalities. And this is a complicated table, but I want you to focus just on the green line. So the green line says that for each of those categories, we saw 100% concordance as defined by the criteria in the study. The arrows and asterisks I'm going to explain in a lot more detail, but essentially that points to additional findings that we saw. So here we see those 5 categories again, color-coded, and you can see the individual numbers of each one of those in this pie chart. The important thing is that we saw an additional 11 cases, and since we had 100 samples, this works out very nicely to 11% of cases where we saw additional clinically relevant findings that had been missed by routine methods. So specifically, 2 of the 9 cases that were reported with complex abnormalities showed additional findings. 2 of the rearrangements cases had additional findings. 4 of the aneuploidy or partial aneuploidy cases had findings. And even more exciting, 3 of the cases that were originally diagnosed with normal karyotypes actually turned out to have cryptic translocations. One of these involved a MECOM ETV6 rearrangement, and in 2 other cases, we saw a 511 translocation with the NSD 1 NUP98 translocation partnership. Going above those 11 cases, we saw that optical genome mapping refined the underlying genomic structure reported by traditional cytogenomics in another 13% of cases. We can also take this data and focus on the additional utility with respects to normal karyotypes, and those are the ones that I already spoke about. But if you break it down as a percentage of the normal karyotypes, there were 48 of them. So 3 of them were reclassified with these cryptic rearrangements. As I said, those were the NUP98 and the one with MECOM rearrangement, but that actually works out to about 6.25% of normal cases, and that's really, really impactful. On the other side, if we focus strictly on the cytogenetically abnormal karyotypes, in 40% of cases, optical mapping better characterized the genomic aberrations by refining the structural breakpoints and identifying unknown cytogenetic elements like marker chromosomes or derivative chromosomes, and I will show you some nice examples of that. Now, not all of those are obviously clinically relevant, or at least using criteria that we know right now. And so we focus really on those that are deemed to be clinically relevant. We see that 8 of 52 cases, or 15.4% of cytogenetically abnormal cases, yielded clinically significant findings. Okay, so if we think about this again in 3 different categories, just simply identifying clinically relevant changes not apparent by routine cytogenomic testing, there were 7 cases, 7 out of 100. Then you had additional findings as well as the potential to refine cytogenomic breakpoints and resolving unknown cytogenetic elements, meaning that traditional cytogenetics showed you something, but we have the ability to further refine them and find additional clinically relevant information, and that was in 4 out of the 100 cases. So there's the 11% total that I showed you in the previous graph. In addition, as I said, there were 13 cases that simply refined the cytogenetic breakpoints and resolved unknown cytogenetic elements. And I'll show you that. So starting off with that first category, identifying clinically relevant structural variation or structural rearrangements and CNVs not apparent. Here we see the 7 findings. There we see 2 of them being the NSD NUP98 rearrangements. And importantly, and this is highlights the subjective nature of chromosome testing, especially when we deal with bone marrow preparations that don't give us the types of resolution that we're comfortable with, in our pediatric cases, really around 350 to 400 band level. Here you see that 1 case involving an inversion 16 was missed with the classic, MYH11 CB FB translocation. This is a case that I'm going to really end off with much later on, showing you a cryptic rearrangement as well, involving a loss of chromosome 5q. Here we see additional information over and above the ability to refine cytogenetic breakpoints and resolve unknown cytogenetic elements. And this was the 1 case that I showed you. It did have a normal karyotype, but as part of the normal workup, FISH was done, did identify a MECOM rearrangement, but in this case, it did not give you the translocation partner being ETV6. And then finally, here we just saw cases where we resolved unknown elements, and I'll show you, as in marker chromosomes or derivative chromosomes, and those are the cases highlighted in bold and maybe a little bit to see in the table. But the important aspect of this is, at minimum, this may just serve as additional markers when we are tracking minimal residual disease. My suspicion is that we will start to see added clinical significance of those. This is a new technology, so these types of things haven't yet been prospectively followed, and that's a lot of what we will be doing futuristically. So to show you some actual cases, this is one where there were no apparent rearrangements in the circus plot. There are smaller intrachromosomal events, as you can see from, the color-coded dots, but they were further reviewed for significance, by overlapping with many of the variants that we are familiar with that are polymorphic and benign in constitutional cases. But after doing that, you do see a deletion in this specific region. So here you can see the patient's DNA fragment in blue, mapping to chromosome 4, showing a deletion of TET2, a very nice example. The classic inversion 16, I cannot help just to show how beautiful this looks. There you see the circus plot starting from P to Q, that's the centromere in red, and then continuing from Q all the way back to P. And there you see on the circus plot a rearrangement between the short arm and the long arm. You can see that there's the patient in blue tracking to the reference as shown with the red arrows stopping at 16p13.11, dissecting MYH11, and then same patient fragment continuing at a different region of chromosome 16 at 16q22.1, dissecting through CBFB. And there you see the confirmation or the same results seen by FISH with the fusion very nicely. We did see additional findings by optical mapping. And again, this is the significance that will be determined, in prospective cases, as well as a very nice large deletion. So if we take the original cytogenetic results as shown over here, as ascertained by karyotyping and FISH, and we add on the optical mapping, here you can see that there is a change in refinement where we did see, an additional resolving of a translocation with better information as well as a deletion. This case really shows the beauty of resolving the clinical karyotype with optical mapping data. And what I'll do is show you the cytogenetics, which is over here. Let's break this up just for ease and clarity and show you what the overall result looked like. So it's quite complex, as you can see, with multiple rearrangements and gains and losses all over the circus plot. But let's start off with the low-hanging fruit, and then you see a gain of chromosome 8. So you see a gain of chromosome 8 in the karyotype, and that's clearly seen even on the circus plot at a very high view with a gain in the copy number algorithm clearly illustrated over there. The deletion 5, again, if we look at even at the algorithm, the copy number algorithm, it shows you a loss of 5q. It's very easily seen there. And the nice thing about the Bionano, they've now worked on a very nice copy number algorithm, very similar to that of microarrays, where you can see the 5q loss over here and the whole gain of chromosome 8. So that leaves us with the additional material on 17, the loss of 20, and the additional material of 22. And you can straight away look at chromosome 20 and say, well, there's a loss of 20, but it's not illustrated on the optical map, the CNV like data over here. That looks normal. And even if you look at chromosome 20 over here, there's some small little gains and losses, but it certainly does not reflect a loss of the entire chromosome. So how do we resolve these? And what is the additional material on 17 and on chromosome 22? So let's focus on those 3 chromosomes. And let's start off with the add 22 at Q13. So at Q13, right at the distal end, we can in fact see there's a small little deletion, as well as a rearrangement between 20 and 22, which means that this whole region of 20 is now sitting at the end of 22. So that's our first resolution that says, yes, in fact, the add 22, the additional material is in fact this whole region of 20. So it is now a derivative 20 with a rearrangement between 20 and 22. We do see a loss on chromosome 20, which translates to this region. But again, this would not explain the whole minus 20. So let's get back to that in a second. Okay, moving on to the add 17, we see that there is a deletion of 17p. We now see there is also a rearrangement between 17p all the way to 20 at p11.21, involving this whole region. So that region now is sitting on chromosome 17, and this is further seen or ratified at the molecule level, where the patient's DNA maps for the first region to 17p, stops at p13.1, and then continues on chromosome 20. So you see direct evidence of that rearrangement at the molecule level. Again, that gives us a great explanation for the add 17. So the additional material from 17 is in fact this whole region of chromosome 20. So what about the rest of 17? We do see it's a lot more complicated. Here we see a deleted region over there, as well as a small gain, as well as a deletion. But what about the loss on 20? So if we break down 20, we've already given an explanation. This part of 20 is sitting on 22. So it's been repackaged away as an individual chromosome, as has this region, to 17, and this middle region is in fact lost. So that leaves that. So what has happened to that region? And if we fill in the full rearrangement, you actually see that those regions have in fact been repackaged on into chromosome 17. So that really gives a great explanation that chromosome 20 isn't really lost. It's primarily being repackaged, and it explains the additional material on 17 as well as chromosome 22. So to end, I just want to show the benefit and that of optical mapping, in that sometimes you may actually have very large rearrangements that are in fact cryptic, and that kind of sounds like an oxymoron. If you have a very large rearrangement, how could it be cryptic? In other words, subtle. And this came from this case where a very large derivative 5 resulting from a rearrangement between chromosome 4 and 5 was not apparent by cytogenetics. And the reason this is important is because it in fact would result in a loss of 5q, which we know does have clinical significance. So here you see the original karyotype, where the only thing that was diagnosed, which was important, was the H21 translocation. But when you look at chromosomes 4 and 5, they in fact look relatively normal. And I think you'd be very hard-pressed for anyone to say that this looks like there is a derivative chromosome. There are two possibilities. One is that this in fact is normal and that the rearrangement we saw is a subclone. We haven't been able to yet go back to the original metaphases and by FISH look to see if they're two subclones or not. But it highlights the issue. And what you will see is that there is a rearrangement between chromosomes 4 and 5 as detected by optical mapping. And what you'll see, this was confirmed by by microarray. Here you actually see that there is a loss of this whole region on chromosome 5, and that's replaced by this whole region on chromosome 4. So you do have an effective loss of 5q, which would be clinically significant. But the reason it's cryptic is that most of us who look at chromosomes routinely know that chromosomes 4 and 5, the banding material is relatively similar, particularly when you have banding levels of 3 to 400. And in this case, you have a region of almost identical size replacing the normal region of chromosome 5, which is why we could regard this as being cryptic. So overall, we saw many cases where optical mapping resolved the karyotype, gave us additional information, and part of the exciting forward-looking studies that we will be doing is to try understand the significance of all these new findings. So as a general summary, optical mapping is a powerful new cytogenomics tool for assessing chromosome structure and copy number. And I think this is the second day of this conference, and I think that is already highly evident. We see, at least in the study, 100% concordance with traditional cytogenomic analysis for karyotype, FISH, microarray, as well as seeing additional clinically useful information that we cannot discern by using routine cytogenetic karyotyping or FISH. It has the ability to resolve complex rearrangements observed by G-banding. It can reveal, the partner translocations, which in cancer is very important for identifying those chromosome gene fusion partners. And these are some of the examples. And I highlighted the MECOM ETV61 in this presentation. And it certainly provides additional new information not revealed by standard cytogenomic workup. Specific to this study, we see that optical genome mapping is equivalent in its diagnostic scope compared to routine cytogenomic methods. It added significant new information in upwards of 11% of cases, with a potential increased diagnostic yield of 6.25% in cases reported with normal karyotypes. If we focus on abnormal karyotypes, in 15.4% of the cytogenetically abnormal karyotypes, it has the ability to reveal clinically significant new events. And as high as 40% of those cases show new findings. And again, that will be part of the future studies to see what they are. And as I said, at minimum, are these effective in helping us track minimal residual disease? So OGM provides a more comprehensive genomic analysis for patients with AML. It has the ability to replace 3 independent assays. You have the karyotype, FISH, CMA. We can detect all those abnormalities at once. This really represents a significant time and cost-saving potential for clinical AML testing. It removes the subjective testing algorithm at individual sites. And this, I think all of us recognize, the way that cytogenetics is done differs literally from laboratory to laboratory, from institution to institution. In many cases, the lab helps to say what should be the next step. In some cases, it's the physician who orders a whole battery of FISH probes and then chooses new ones as new results come up. And so what this can do in one shot is standardize care and healthcare costs. And the most exciting thing that I see this is a potential to remove disparity in healthcare, because many of these decisions are actually based on what a patient can afford or not. In some cases, there are labs that do significantly extensive panels of FISH and karyotyping, potentially even CMA, and that will be covered. But for those patients who can't afford that whole extensive battery, we now have a potential one-step test that can do this all in once and really take away those disparities. So the next step certainly will be the discovery phase. What recurring abnormalities do we see? Can we track minimal or residual disease? What is the prognostic potential value of any new findings? Again, that could lead to potential therapeutic value. And this is all going to take a lot of fantastic collaboration with our clinicians. And so I'm going to end off there. I do want to recognize a fantastic group, as you see over here. I've mentioned the primary individuals at each of the centers at the very beginning, but there are more than just those individuals, as you can see from this slide. And my sincere thanks to each and every one of them. We literally met weekly to discuss the results and update. It's been a 2-year effort. And I do want to thank the Bionano folks for supporting us on the technical side. It's been a terrific adventure. And I'm going to stop there. Thank you very much.
Welcome back, everyone. And first of all, I would like to thank all of our speakers today. Absolutely amazing presentations. And it was a pleasure to see everybody from the United States, from Europe and China, showcasing all the work going on in the laboratories. So we have a number of questions today. And again, I would like to introduce Dr. Sven Bokland, our Vice President of Communications and Marketing and Scientific Affairs. So he will be tag teaming with me as we take this live Q&A. But I do have a question or a number of questions that came up yesterday, which we were not able to address. And I'm going to start off with Bryn. A number of questions came to us yesterday talking about the differences between LDTs or laboratory developed tests and the utility of FDA clearance or 510K clearance in terms of newer technologies being adopted in the clinic. So would you mind commenting on the current technologies that are used as standard of care and what your thoughts are?
Okay, I'm sorry. I'm going to make you repeat that question just because I was transitioning from the two different the live view.
Sure. So the question is that would you mind telling the audience about your thoughts on
Welcome and thank you for listening to my talk. I'm Barbara and I'm supervising the routine diagnostic lab of genetics of hematological malignancies at the university.
I see. Okay. I'm sorry, we had some technical glitches right now. So Bryn, the question is, that would you mind telling the audience the differences between laboratory developed tests and FDA clearance or 510K clearance in terms of the presentations that we saw in constitutional genetic disorders and hematological malignancies, and what the current techniques, which are standard of care, used globally, and how those differences can be understood well by our audience and viewers.
Right. So actually, Alka and I were speaking yesterday, and I said one of the most important things to really realize, at least in cytogenetics, is that the LDTs, the lab-developed tests, are in fact really the norm. It's basically allowed us to get clinical testing to our patients and really give them the best care. That is the norm, and the FDA part is kind of the exception. And a lot of this has to do more with I think the bureaucracy and the time that it takes for the FDA, approved tests to be available. There are advantages for the labs to have FDA-approved tests, and a lot of it has to do more with reimbursement and less about the clinical utility. And so the pathway that clinical labs have chosen, at least in the United States, of following appropriate clinical validations in their labs as well as in various studies, is to say, okay, we can't wait 2-3 years for this now to be approved by the FDA. We want to be able to offer this to our patients now, and we believe there is enough clinical evidence that exists in order to do that. And so the LDT pathway has been the mechanism that the clinical labs have chosen to take, tests that have come from a research protocol and been shown in a clinical research fashion to be applicable and relevant to bring those into clinical testing for their patients.
Absolutely. Thank you so much, Bryn. And I think that this question was really relevant for the US audience because, in Europe and rest of the world, this is probably not applicable. Sven, would you take the next question for one of our speakers, please?
Sure. I think that's Barbara Dawala who joined us as well, right? Barbara, you gave a nice comment all the way at the end of your talk where you explained that you are now actually implementing optical genome mapping for routine diagnostic use in Belgium, that you're going to reduce FISH to 0 or 1 tests per sample, reduce PCR. Can you tell us something about this process, how that is working for you? Are you implementing this workflow for all your ALL samples, or what part of your samples are you now going to analyze with Saphyr?
Yeah, we do it for all diagnostic ALL samples. Yes, we will implement it for all of them. Also for the adult.
Barbara, we're having some audio issues, I think.
Okay.
Yeah, now it's better.
Okay, so we are indeed implementing it for all the diagnostic ALL cases, both for the pediatric and the adult ones. So we are really changing the workflow for all the ALLs.
And that has already happened, or that is something you're implementing in the future?
We are currently implementing it. So the new ALL cases that come in, the two came in this week. So we are analyzing them with Bionano, and we are also still using the conventional karyotype. But yes, this is right.
That's incredible. Yeah, thank you.
That is absolutely incredible. Thank you, Barbara. It is so nice to hear that you are using this tool in the clinic after completing your successful validations. Thank you so much. Our next question is for, do I see Valentin? Yes. Thank you so much. You're, again, for an amazing and incredible presentation. One of the questions was that on your optical genome mapping findings, the unique findings, when you did all the comparisons, Would you comment on the clinical actionability of some of the translocations and additional findings that you found by optical genome mapping? I think we again might be having some audio issues. I apologize. We do have all of the presenters and all the speakers live. So maybe we can come back to Valentin when we can hear him. So let's go to Rashmi. Rashmi, you showed an enormous amount of data on myelodysplastic syndrome cases and on 50 cases. One of the questions is, could you speak to the relevance for some of the audience on the relevance of IPSS scoring and how that is important and how these additional findings could really help in the stratification of these individuals and patients affected with MDS? I thought I saw Rashmi a few seconds ago.
Let me maybe go to Gordana in the meantime.
Yeah.
Gordana, you showed several examples of patient samples that you were able to analyze with optical genome mapping that failed with karyotyping because the cells don't culture, and therefore maybe give you a skewed view of the genome because you're only seeing a certain type of cells. How big of a problem is this in general, and how much do you think optical genome mapping really is an advantage there?
Gordana, I think you're on mute. Let's see. William, is there something that we can do to unmute Gordana because we can see her?
Maybe in the meantime, we know that Bryn and Barbara's audio work. Do you have any thoughts on this? How big of an issue is cells not growing in culture, and how much is optical mapping a solution for that?
Well, if I can talk about ALL, this is really an issue. So we recently calculated our numbers for the year 2020, and we had about 10% of cases that didn't want to grow in culture. So yeah, it is actually a problem in ALL. So there, Bionano can be really useful to help. So sometimes you have really a failed karyotype, but you can also just have a normal cells and not the abnormal ones. So not really the tumor cells, but normal cells.
I think we're all having some echo. Thank you, Barbara. I think we have Gordana back now. Gordana, do you remember the question?
Yeah, I hope that people can hear me now. I don't know if it's better.
We can hear you, but there is certainly some background.
Yeah, I'm not sure how to deal with the background, but just want to second what Barbara just said. That especially in BLL cases, it's known that not only significant percent does not grow, and about 10% is our estimate as well, but also morphology is really, really poor. So even those that do show you abnormalities by cytogenetics, it's very difficult to characterize them. So I see huge value in optical mapping that obviously does not depend on cell growth and culture.
Thank you, Gordana. I see Valentin is back on. So, Valentin, I don't know if you heard the question earlier, but the pathologies, you had some cases where you had clinically actionable findings. Would you comment on those, the additional findings, and how they really impact the patient's diagnosis? Let's give it a minute and see. Okay. All right, Rashmi is back on. William, Sven. Sven, we can't hear you now.
Hey, Rashmi. Thank you for joining us as well. You talked about how this IPSS scoring really determines treatment and prognosis in MDS cases. Can you talk a little bit about how important this really is and how a structural variation method that shows you more of these variants can matter there and potentially change how you are treating your MDS patients?
Sure. I hope you're able to hear me.
Yes.
Okay, perfect. So RIPSS, it's a prognostic scoring system. It's really the basis for management of any patient with MDS. The first step is the risk stratification, and these include decisions whether to start the treatment or just manage the patient based on observation. Or if the patient is already on treatment, the decision to modify the therapy. Everything is dependent, at least at this time, it's based on the RIPSS score. And there are three components to it, in addition to the bone marrow blast percentage and the degree of cytopenias. The third, the most important component, are these chromosomal aberrations. And traditionally, until now, it's only been based on what we can interpret from conventional cytogenetics. And with these novel technologies detecting many cryptic aberrations and much larger aberrations that are possibly missed by just morphologic evaluation under light microscopy, I think there is a lot of interest to look into this, how these aberrations actually change the risk stratifications. And the outcome of MDS, it's very heterogeneous. Even patients with diploid karyotype, some of them don't do very well. And the one possible reason might be these abnormalities. We are just not detecting them. We are just falsely calling them as diploid because we are missing them. And I think optical mapping has a very important role to play in this.
Thank you, Rashmi. Thank you so much. Excellent answer. So our next question is for Tomo, and I hope that we can hear you without any glitches. So in my introduction, I had shown about the variant classes detection, and right now that we are working on the ability to pick up LOH. But Tomo, you showed in your presentation that your team has an algorithm to accurately detect copy-neutral LOH using optical genome mapping data. Could you please let the viewers know that along with all the other gamut of all the structural variants, how this is useful and important?
Yeah, so I think for the copy-neutral loss of heterozygosity, there is not like a ready-made algorithm by Bionano or so. So we kind of just did it manually here. So we actually had to rely on the de novo assembly-based tool to get the zygosities for the actual structural variant calls. And based on the zygosities, we just manually looked if there are large stretches of chromosomes that only have homozygous calls, and we just did a simple manual work on that. But that, of course, has some limitations if the sample is very heterogeneous, or the tumor cell content is very low. So then you cannot really rely on the zygosity call only. But this was a really clear-cut sample that we were able to do this for. But I think that's really important. One step that needs some kind of development also from your side would be really nice to have it like a built-in for the software. Yeah.
I can comment on that, and I promise you that we are working on it. And so, we are looking forward to working with your team on that. But as a follow-up to that, there are a number of questions on the clonal diversity and the comparisons that you did with the clones and the allele fraction as limit of detection. So would you just briefly comment on looking at the data and looking at all of the maps, how those results were concordant in terms of the clonal evolution of these specimens?
Yes, so we used a cutoff of 10% of variant allele fraction on the samples that we analyzed. So we were not really we kind of knew that the limitations around there. So anything below 10% we ignored from the current study. So and the samples we had were around 300 with the current coverage that we are using. But with more coverage, we could, of course, pick up events that have less than 10% variant allele fractions, and that would be. But overall, the comparison between CMA and Bionano, we did that for the variant allele fractions, and those were really well concordant between these two approaches, actually. So there was no big surprises in there. So we were happy with that. Yeah.
Thank you so much, Tuomo.
Yep,
Sven,
sure.
our next question.
We got maybe for Bryn, it was a question for the whole team, but how do you confirm any of these newly detected translocations that you find with optical genome mapping with other techniques? I know, Bryn, that you have done so. Maybe everyone else can comment on that as well.
So Bryn can start, and then we will open the floor for other speakers so there is no overlap.
Yeah, so we've done that. Jim Broach did a lot of them for the cancer. I can tell you that at Columbia, we've designed kind of a custom Sanger across the breakpoints. It was something that we spent a bit of time on, and we've done a few cases that way. But I think it's worthwhile investing in, because ultimately we're going to see a lot of new things. And so I think that if you are going to use this, you should have some type of validation follow-up ready to go, at least, while it's still considered new technologies. I don't know what everyone else has been doing.
Thank you, Bryn. Valentin, let's see if we can hear you now.
Ready to go, at least, while it's still considered new technology. I don't know what everyone else has been doing. Thank you, Bryn. Valentin, let's see if we can hear you now. Ready to go.
Jan, I see that you
am the
want to answer it.
Maybe I can speak for him if he doesn't. So, can you repeat the first question you had for Valentin, please?
The first question was, you had some very unique findings, structural variant rearrangements by optical genome mapping, and the question was regarding the clinically actionability of some of those findings associated with the leukemia.
Okay. The new variants we have found had a prognostic value, but a weak prognostic value regarding the other rearrangements that have already been found on those patients. So it did not change the way the patients were treated in that 10 patients.
Mm-hmm. And thank you. And the second question right now is that when you have additional unique findings by optical genome mapping, are you using orthogonal methodologies for confirmation? of those that were missed by the standard methods, or going back to using, for example, as Bryn was talking about, spanning the breakpoint with PCR, or trying to use some FISH rearrangement, other probes, as other speakers showed?
Yes, we could confirm some of the alterations by going back on the raw data of SNP array. Some abnormalities, in fact, were visible in the raw data, but had not could not pass the filters in SNP array. And some of them could be targetable by FISH, and the last ones will be checked by PCR and expression, but it's ongoing for this time.
Thank you so much. Anna, let's open the floor for you. Would you like to comment on this last question too? So we'll just take it with all the speakers. We cannot hear you, Anna.
Do you hear me?
Yes, now we can.
Okay. We haven't validated any abnormality yet, but we are planning to validate by FISH, by chromosome painting, or with non-commercial probes with bacterial artificial chromosomes. This is our idea.
Perfect. Thank you. Rashmi? We again cannot hear you, Rashmi. Okay, but let's go to Barbara while Rashmi comes back.
Yes, so in Leuven we have a large panel of FISH probes that we use to confirm the extra aberrations. We also use lab arrays or low-pass sequencing to confirm some of the aberrations, and we are also performing RNA sequencing for all the cases. That helps us to confirm the extra aberrations. So yeah, we use, depending on the type of aberration, different kind of techniques.
Thank you, Barbara. Tomo?
Operations and those we were able to find back, but the next step is, of course, to understand what else is there. And we have done some validations for samples that we already had, like short-read whole genome sequencing data available, and some FISH validations as well. And overall, it seems that the at least for like simple deletions and duplications, the short-read genome sequencing, you can confirm even most of the extra events that we saw with the Bionano. So overall, but we have just started with the orthogonal validation, so we are not very far with that actually. Yeah. But,
thank you so much, Tomo. Rashmi, we can read your messages, but we cannot hear you yet.
I'm not sure what happened though.
So do you mind commenting, because I think we can hear you, and then we'll go to Gordana.
Oh, okay, yes. I'm not sure what happened. Sorry about that. But yes, the samples from the pilot study, we have confirmed all of the additional aberrations, mostly using microarray, and for the fusions using FISH. And then we did have one deletion that we could not, it was a really small deletion that we were unable to pick up by microarray, and we are doing the long-range PCR to confirm that deletion. And we also confirmed a couple of fusions using an alternate, a targeted RNA-seq fusion assay by another company.
Okay. Thank you, Rashmi. Gordana, I think I saw you. And then do not see you. So I think the viewers, probably the audience, was interested in seeing how the speakers and the users across the globe, the different orthogonal methods that they are using when they are showing all this data about clinically actionable and additional findings and drivers playing a key role in the pathogenesis of these heme malignancies. So this is all that we have time today for. I mean, it went a little bit longer, but I think we have to congratulate and really applaud all of the speakers, and thank you for staying till the end. And I hope to see you all tomorrow morning again for our solid tumor session. Thank you all.
All right, so I just want to check if you guys can actually hear me. I'll be holding another live session on tomorrow's presentation, of course. And this is the symposium, of course. I'm going to bring up the chart so I can show you my chart analysis of the stock. And we're also going to talk about a mistake that I made in my last live session where I was talking about the FDA approval, and I'm actually going to clear that up for you guys because there was mention of the FDA, but since I wasn't searching the actual name of FDA, I was just searching the acronym. I wasn't able to find it, but I'll pull that up for you later after I pull up Thinkorswim. All right. I just want to make sure you guys can hear me. Type something in the chat if you can hear me. Can anybody confirm that they can hear me? Hello. All right. Come on now. All right. I'm going to have to restart Thinkorswim because I'm having a couple issues with it. The problems of having a Mac, but so I'm going to pull up the SEC filing first since Thinkorswim wants to act up on me and show you guys what they said about FDA approval, and this was filed on August 25th, 2020. So if we search for drug, no, federal securities, no, not seeing it in this prospectus. U.S. federal income, this is not it. Let me check the other prospectus, see if I'm missing anything. Federal drug, no. Okay. Well, this isn't going as planned. Okay, so it might be in this prospectus. Just give me 1 second. Pull this up. Let me also pull up the live stream. So, okay, not seeing it here either. Anyway, I'm gonna have someone pull that up for me in that recent prospectus. Give me 1 second. All right. So pulling up the price charts now, we'll wait for it to load. So right now they're kind of hovering up and down between this 546 resistance level as it stands now, but of course if it breaks through this, it could go all the way up to this next resistance at about 590. There's also a resistance somewhere around this point as well, around 564 or so, that it's going to have to pass in order to get to that 596 mark or so. So this is the price that I'm looking at. So this is the 2-day 5-minute chart, and I started basing my price analysis a couple of weeks ago whenever they first caught steam. Also, I want to point out that they just got saved from being delisted by keeping their share price above $1 for 10 consecutive days. So, I mean, unless it drops to $1 from here, which I don't think that it will, that would have to be like an 80% drop, which would be absolutely insane. Okay. But basically, if we go back and look where I started this price analysis on the 10-day and 30-minute chart, I believe, right here, you see I started my analysis based on this, where we saw a peak in volume, and then it shot all the way up. And of course, there was also a support somewhere around here, which they broke below. And during their symposium, it's been doing kind of a whole lot of nothing in comparison to what they were doing a couple weeks ago. And this, I think, is mostly attributed to a lot of investors not truly understanding the science behind it. And while there is quite a few knowledgeable people here in the chat, I think that the general consensus is that it kind of went over a lot of people's heads. And that's kind of to be expected, because science is hard, and I'm not going to pretend like I understand the science behind it. That's why I had MKC as a moderator in the chat to try to clear up any questions that you guys might have had. So I'm going to try to search for that prospectus one more time and see if we can pull that up, because I really want to show you guys FDA approval and their stance on FDA approval as of right now. So let's see, prospectus. And if you guys want, I will link this to you. And I'll pin that in the chat for you guys. Prospectus, I think it was in their most recent prospectus filing, but I'm not 100% sure. Food and Drug Administration. I keep thinking that it's Federal Drug Administration, but silly me. Food and Drug Administration. Okay, so I don't think that is in this one, so let me check my other sources. Let's see. This is not. I really hate the new version of Mac OS, but I'll have to live with that. Let's see. Alright, I'm just going to go back to the share price since I can't find this information right now. So right now we're trading at 541. And if we zoom into the 2-day 5-minute chart, yeah, I did lose the filing, unfortunately. If we go back to the 2-day 5-minute chart, still right at that resistance line. And if we get a break through it, then again, we could go up. But of course, it could always be rejected like it seems to be getting now. But it could always go back up. Stocks go down, up and down by the minute. So, and if we continue to go down, not sure why it's drawing price lines for me. But if we continue to go down, I see it probably hitting 510 or so, and then possibly falling all the way down to 491 on the downside of things. So, also, there was mention of ARK Invest buying into the stock somewhere in the chat, and I think you guys kind of linked the timestamp to where ARK was mentioned in this live stream. I don't know how long ago it was at this point, probably around 20 minutes or so. But they did mention that ARK is in talks with this company, but that doesn't necessarily mean that ARK is going to buy, because Simon from ARKG and all of the other sort of researchers or analysts for ARKG, they're always in talks with genomic companies all the time to try to get a feel for different companies and the sector as a whole to see where they might want to invest in, but that doesn't necessarily mean that they are going to invest into BNGO. And it could be the case that ARK does decide to buy, and that's really up to them. And I left a resource for you guys that you can check out where the resource where you can actually see what ARK is buying and selling, and that's going to be in their email list. How long do I think it would take for BNGO to hit $6? I mean, it's really hard to tell like when it could happen. I mean, I don't have like a crystal ball or anything, but if it does go to $6, I would think that it would happen by the end of the week, but really there's no guarantee of that actually going through. It could always go down. People might not like the symposiums as they were expecting, but I think that you guys seem to be enjoying them quite well, and hopefully you're gathering more information on whether or not you want to invest into the stock. And that's what I'm trying to really provide here. Again, I just want to state that I'm not long or short on the stock. I don't have any position. I'm just trying to share with you guys the information that they're putting out. That way you can make a decision on whether or not you want to buy or whether or not you want to sell. It's all up to you. Just wanted to get that out of the way. Let's see. ARK is dumping PacBio. Well, ARK is always buying and selling new stock. So that's kind of the thing with them, and that they're an actively managed fund, which means they can buy and sell whenever they want. Meanwhile, things like the S&P 500, which is made up of 500 different stocks and is seen as kind of the epitome of American investing, they can't really decide, oh, I want to sell this day. Meanwhile, ARK can really do whatever they want. At least in that matter. And usually they have about a 5-year investment horizon that they look at whenever they're analyzing a stock. But of course, they're always subject to sell out of some of the position if it starts to make up too big of a position in their entire portfolio. So that's something that we've seen with Tesla, and I'm not trying to make the comparison between the two stocks, but ARK has been selling some of Tesla as it continues to rise, just to keep them under the 10% barrier that they've kind of set for themselves, basically. Yeah, ARK does rebalance daily, and of course, you can see whatever they buy or sell in their email list. Let's see. I'm going to try to find this FDA. I'm going to look at this file one more time, see if I can actually find this dang thing. There was mention of the Food and Drug Administration, the FDA, but I'm not sure exactly where it was, and I can't do Control F or Command F in this. So let me actually pull up the link for it. Let's see. Oh, where am I getting that noise from? Sorry about that. Okay, so now I should be able to search for it. Food. No, I'm still not finding this. Okay, so I'm probably going to make a community post on this once I can actually find this information. But basically, from what I've read on one of their latest prospectus, they said right now it wouldn't be great in terms of money and time in order to seek FDA approval or to actually get FDA approved, because it is a very time consuming and very expensive process to go through. Let's see if there's any questions in the chat. Next thing, when do we find out if it's going to be approved? It really depends on whenever they applied and just how long it takes them in order to do it. And honestly, you really can't judge it based on other companies. I know a lot of people have been saying that, well, somebody in the company, I think it was the CEO, has worked for another company that got approved by the FDA, but you really can't make that comparison just because they have different technologies, and FDA approval is based on how your technology works. So let's see. Let me pull up the live chat one more time, see if you guys have any questions. Want to look at the price charts again, see where it's moving. So it seems to have been rejected by this 546 level. It could be heading down to about 530, possibly all the way down to 511, or maybe if Cinemet continues to be bearish on the stock, about 491. Currently, they're down 5.3% of the day. So it's not an awful lot considering how much it rallied all the way up to 638 on Monday. But you do have to keep in mind that stocks do go up and down by the day, by the minute, by the hour. And I mean, it's really subjective to market sentiment. I really appreciate that, Kumar. Got in at 540, give up 740, don't regret. That's a really good profit if you actually got through with that. That's really, really good. And I know a lot of people bought in at like $1 or like $3. Depending on how early you got in, that's kind of going to determine how much profit you take out of this, unless you are planning to hold it for the long term. And there's definitely some room for growth for the long term for BNGO, just considering the genomic space. But you also have to keep in mind the reach that their technology has. So when people compare it to BNGO, who has multiple technologies that do genome sequencing, BNGO only has a single technology, and that is used for genome mapping. So those are kind of two completely different processes that you have to keep in mind whenever you compare the two of them, basically. That's what I'm trying to get at. So that's that. Let's see if you guys have any other questions. The public offering. So public offering is that could negatively affect the price in that the people that they're offering it to through writing options, they're buying or trying to buy for what is it, like $2, I think it was. Somebody confirmed in the chat what price that it was. I'm not 100% sure. But that public offering kind of sent their stock price down. I don't remember when exactly that happened. I think it was sometime right here. I think that was announced whenever they fell yesterday. let's see, 305. Yeah, I think you're right, TQ. I think it was 305 or somewhere around that price. I think you can actually look it up on the SEC website. We can do that here if you guys like. SEC.gov reports. And we'll search for BNGO, of course. So let's see. That would be in there. Let's see. It's either in their 8K or their S3 filing. I'll have to check into this. Current report 305. So this is the information you need to know about the public offering. The price of the public offering is $3.05, and the underwriters have agreed to purchase the shares from the company. So this isn't coming from a shareholder or inside shareholder dilution, but it's coming from the company. And this is pursuant to the underwriting agreement at a price of 2.867 per share. And this is an offering of 4.3 million shares. Also, something that you want to keep in mind with this stock in particular is the holding and all the holdings and how it's separated. So what I've seen in my research on this company, about 5.4% is held by insiders of the company. Then I think about 6% or like 5.6% is held by institutional holders. That's going to be your JP Morgan. It's going to be Vanguard. There's a few others, and I can actually show you that too. We can detail the holdings of this in today's live chat just to show you guys what exactly is going on here. Let's see. BNGO institutional. So this is the institutional ownership for BNGO. So this is, again, going to be Vanguard. You have Bank of Julius Baer. You have Geode Capital, Perkins, and you can also see the values of how much they own, and this is going to be in thousands. So all of these companies right here own about 5.64% of the company. And that's also subject to change as they prove themselves in this market and become more established in the genomics field. When do I think the option chain will be included? I think that I really couldn't tell you like a date when that would happen. In particular, I can't like say, oh, it's going to happen next week or it's going to happen next month. I think that's really up to the SEC to decide on that, and you might be able to track the SEC filings to see when that might happen. We can try to find it in this SEC filing to see if they have any mention of opening the option chain. So the underwriters are granted a 30-day option to purchase up to 4.3. So they're writing it through options, but that doesn't necessarily mean they're going to open an option chain. I don't believe, but don't quote me on that one. So the exception for issuance of options or restricted stock in the ordinary course of business since the respective dates. The company has not entered into a granted convertible or exchangeable securities, options, warrants, agreements, contracts, or rights in existence to purchase or acquire from the company any shares of the capital stock of the company. So I don't think there's anything, actually right here, stock options, the exercise of each price of each call option under the company or employee has been no less than the fair value market of the common stock is determined on the date grant of such option. I'm not sure. It doesn't look like to me that they're going to be open an option chain on this. So I hope that kind of answers your question. to some degree. You could also look into these SEC filings and try to decipher for yourself, but this is just at least what I'm getting out of it. Let's see. Let's check out the price charts. Again, really their stock isn't moving based on the symposium. I think a lot of people were expecting a lot more out of their share price, but it seems like it's kind of going over a lot of people's head. And, you know, it's kind of to be expected, as I've said, whenever they're using big science terms, showing all the data. I mean, I really can't understand it. So I try my best to wrap my head around all of this, but I mean, I work with stocks. I don't work with science. I don't work with genomics, so keep that in mind. Prediction up or down for BNGO tomorrow? I mean, really, flip a coin, it could go either way, honestly. It really depends on what they present, how investors feel about what they present, and if they're willing to put their money down, basically. And if they're not, of course, it's subject to go down. If people are more willing to sell out, then the price is going to fall. It's all depending on supply and demand, of course. Whenever you have more demand for the stock and everybody wants to hold it, you have the price go up. And when you have more sellers and less people buying in, the price comes down. So it's really just subjective to investors' perspective on this. Look out, Louie. We'll be there soon. That was a nice little rhyme. I like that. Anyway, so I'm going to be wrapping up the stream in about 13 minutes. I'm going to do it in this at around 2 p.m. And if you guys have any questions about the share price, about this stock, maybe the SEC filings, if I can try to sort through those, just let me know in the chat section, and I'll try to answer them to the best of my ability. So yeah, I do think that people want to come up with a conclusion after all of the symposiums have been completed, and that could be a catalyst for the stock to go up or go down, depending on people, if people really enjoyed it and if they understood what's going on. Yes, Ismail. Sorry if I botched your name. What do I think about ZOM? I haven't really done too much research on ZOM, but something that I did want to cover that I mentioned earlier that I would talk about in my live session is a couple of speculative plays. So currently, I see BNGO as a speculative play, in that I'm not sure if it's going to have as broad of a reach as many people expect of it. So a couple of the speculative plays that I have right now in my portfolio, which I'm very limited on them. I have SSPK, which is weed maps, and this is basically the Amazon of cannabis, in my opinion. And I bought in. I don't know if you guys watched my video on this, but I bought this stock at around $12 or so, and I'll show you on my Robinhood account just to show you that this is real. But I bought into SSPK a couple months ago, or maybe like a month ago, probably around 6 weeks. I really don't know, and I can't keep track of time at this point with quarantine and everything. So, but SSPK. So, I bought into SSPK, which I think is the Amazon of cannabis, and I bought this at $12.35. And I still believe in the company. If you look at their investor presentation that they presented whenever they announced their merger, they have really, really good financials. And that's what I love about this company, and that they're just continuing to expand the revenue more and more and more each year. And then I want to show you guys the other speculative play that I'm in, and that is going to be, give me 1 second. I just bought it yesterday, so give me 1 second to scroll through this. Also, I just want to say that I'm not a financial advisor. I'm not a professional at all. So this is not advice. This is just what my personal opinion on these two stocks are. So I bought into FLGT. So this is another genomic stock. It's called Fulgent Genetics. And I bought yesterday. I bought only $100 because I haven't put in a ton of time and research into this company yet, but I am planning to put more effort into it. So right now I only have $100, but I'm planning to build this position up to maybe 1 to 2% of my account, which would probably be around $400 to $800. And I think that this company has a lot of long-term potential, considering its market cap of $1.8 billion, and they're doing a lot of great things, at least from what I've heard. And we actually gave a breakdown of what they're doing in our premium Discord that you can find in the description of this live session. But I think that Fulgent has a lot of potential, and I'm not trying to say that BNGO doesn't, of course. It's all really up to your perspective on how you see everything unfolding with genomics. But right now, at least, Fulgent is my genomics play. And I also have ArcG, which I think is the safest investment whenever it comes to genomics, because they put in all the time and research for you. Meanwhile, you just get to reap the rewards. And I bought this at an average price of $97.75, currently up 7%, market value of $600 almost. So, and if ArcG does decide to buy into BNGO stock, I would be happy with it. But if you guys look through the email list, you'll be able to know whenever ARC does buy and what they sell the date of, basically. What is STPK? I haven't looked into STPK, but I will. Stark Peak Energy Transition. They're up 10% today. Let's look at the price charts. I want to look at what they do first. So they're a blank check company. If you guys can tell me who they're merging with, so this is a SPAC, I'm assuming. If you guys can tell me who they're merging with, what a company is doing, that would be great information to know. That way I can do a better analysis. But if you guys just want to look at the price charts, I can pull that up for you. And right now, according to the 180-day 4-hour chart, they are pretty overbought considering the RSI is at 68. But that doesn't necessarily mean that they can't continue to go up considering how much volume is flooding into it right now. It's been flooding in for the last about 5 days or so. So we can look at the 5-day and 15-minute chart just to get a better view of what's going on here. And that's how I like to base my analyses lately, just to see when the volume started picking up and do your price analysis from there. And we can actually do a price analysis quickly just to wrap this video up. So I'm going to start by creating a trend line at the bottom, and we're going to set, I meant to make a trend line, not that. So we're going to start by making this trend line starting here and then following the volume. So this should act as a support for the stock, seemingly. And I'll actually link this. I'll link this price analysis somewhere in my Discord, which will be. And if we use this as a top trend line, the start to a top trend line, there was a peak of volume here. They were overbought during this period. There was hardly any volume, so you can't really use that as a high. So if we extend this to the right, we can see this is sort of a channel, and that you could see it come all the way up to the top of this channel to 37.67 by the end of the day, or you might see it fall down to 33 32.69. And if we try to do some support and resistance lines, we can find there's one right here at around 35.25 is a resistance line, and we can draw that out. I'm not gonna try to make this chart pretty like I usually do, because that would take too much time. But this could be a support right around here whenever they saw this peak in volume. So right now there's a range that it's trading in, which is between right here. So that is going to be 33.41 and 35.32. And it looks like it's approaching that. So if we see a break through that resistance, we could go all the way up to this top trend line. And if we break through that, we could be heading to new all-time highs by the end of the day. And also, this is all just subject to market sentiment, volume, and how investors are feeling about STPK. What's my end of week or end of year predictions? I'm assuming you're asking about BNGO, but clarify if I'm wrong. So for my end of week on BNGO, my prediction, if we zoom out of this, I think, let's see, give me 1 second. Bear with me here. So the end of the week is going to be the 15th, yeah? So by the end of the week for BNGO, if we base our price analysis from here, maybe they could go to, let's see, by the end of the week, they could maybe go to like 690, or possibly, if people don't like what they see with the symposiums, maybe to 450. But of course, this is all just what I'm seeing. You also have to base this on the price levels, so maybe somewhere in between 424 and 655, or possibly even 714 by the end of the week. And of course, this is all subject to change as the price moves, and I'll probably give you a more detailed price analysis tomorrow on BNGO. When did BNGO said that they're in talks with ARC? I can't give you a timestamp. I'll try to put it in the pinned comment below after this live session is completed. I'll try to sift through it and find that point where they mentioned it. But from what we've seen on Simon Burnett's Twitter thread, it does look like they are in talks with them. And they kind of confirmed it, I guess, at least from what I've heard. But basically, Simon has been giving his thoughts on this company through a Twitter thread that he put out, and this was on January 5th, and I'll check for any updates from him as well to see if he's given any new information on BNGO and their technology. Genome assembly and phasing. Let's see. So I'm not seeing any mention of it lately, but I do think that they are in talks from what we heard in today's live session and what we heard from Simon Burnett in his analysis of their technology. So it is 2 p.m. Eastern Standard Time. If you guys have any other questions, please let me know. You can either leave it here in the chat and I'll get back to you, or you can leave it down in the comments below this, and I'll try to get back to you as quickly as I can. So that wraps up today's live session, and again, I'm going to be holding another one tomorrow since you guys seem to like these live sessions so much, so I want to thank you all for being here. Thank you for your support. And as always, remember to stay positive, stay green. I'll catch you in the next one. Bye, guys.